Bioinformatics, School of Life Sciences, Pondicherry University. Sir obtained the PhD degree in 2003 from Punjab University, Chandigarh, and he has about 9 years of teaching experience. He started his career as a research officer, ICMR, Center for Advanced Research on Standardization, Quality Control, and Formulation of Traditional Remedies, Regional Research Lab, Jammu. Then he moved to Ranbaxy Research Lab, Medicinal Chemistry Division, Gurgaon in 2002, and until he worked up to 2003. And then he received the postdoctoral fellowship at Division of Biotechnology and Pharmaceutical Research at National Health Research Institute in Taiwan. Until 2010, he worked there itself. Then in 2010, he joined as an assistant professor in the Center for Bioinformatics, School of Life Sciences, Pondicherry University. And till date, he is working in the same institute. Sir, areas of expertise are computer-aided drug design, medicinal chemistry, and biochemistry. So Mohan Kumar is an outstanding researcher. It is evidenced by he published more than 80 research and review articles and book chapters, and received more than 1,500 citations and H index of 23. And his papers were published in reputed journals such as Future Medicinal Chemistry, Scientific Reports, Cancer Treatment Reviews, Proceedings of the National Academy of Science, USA, and Journal of Medicinal Chemistry. Sir has co-inventor for 16 international patents. One of his invention is approved by US FDA for testing in humans for the treatment of cancer and undergoing the phase 1 clinical trials in Taiwan for patients with head and neck cancer and EGFR mutant lung cancer. Another molecule is also approved by US and Taiwan FDA in 2017 for testing in humans for treatment of cancer. And apart from this, 3 patents were granted. They are fused multicyclic compounds as protein kinase inhibitors, fused bicyclic and tricyclic pyrimidine compounds as EGFR kinase inhibitors, and fused bicyclic pyrimidine compounds as Aurora kinase inhibitors. These are the 3 patents granted. And Dr. Mohan Kumar received the grants from various funding agencies such as DBT, DST, and UGC worth of around 50 lakhs. And all these above indicates that outstanding research contributions in the pharmacy field, and also setting a new trends in the pharmacy research. He is also the life member in IPA, ISCA, and American Chemical Society member. With this brief introduction, I invite Dr. Mohan Kumar to deliver lecture on CAT techniques for drug discovery and case studies in de novo drug design. Over to sir. Thank you, sir. Thank you very much.
Thank you, Sridevi, madam. Am I audible?
Yes, sir.
Yeah. So, can I go for presentation?
Yes, sir. You can, sir.
Okay, my presentation, I think it's visible now.
Visible, sir.
Yeah.
Visible.
Thank you very much, madam. First, I would like to thank Sridevi, madam, for inviting me for this faculty development program. Of course, previous year also I was there in Tirupati, physically there. But due to the coronavirus, now we are not able to have a direct interaction. Thank you. And also I thank other organizing members for this Atal program. Let me see. Okay, yeah. So what I'm going to, so one thing I learned is like the faculty whoever is attending today is like from pharmacy background. So some of the information that I'm going to talk about, maybe it's not very new to you, but still I will try to maybe give a brief idea of, so my talk is going to be like a 3 sections. First will be the drug discovery process. I will give you a brief view from my perspective, because now basically I'm a what do you call experimental medicinal chemist till my PhD. Then when I moved to Taiwan for my postdoc, I had working with a different multidisciplinary team. We were trying to develop drugs. So I had some experience of interacting with people who work on computer-aided drug design. So till I came into bioinformatics from 2010, typically I am not a computational, or what do you call, I'm not an expert on a computer-aided drug design. But once I move here, based upon my research interest and the experience I gained over here, now most of the time I do work on computer-aided drug design because I'm in bioinformatics. Most of the students here are from biology background, and they have more experience on the bioinformatics side. So now typically I don't work mostly, because even though I did most of my PhD is on the chemical synthesis, even in my postdoc also most of my work was whatever the patent madam was showing us on synthesizing of those molecules. So that was my expertise till 2010. But after coming here, I learned because of my interest and the necessity also. Not only interest, because the students are oriented to bioinformatics. So I could, so another thing is like I try to integrate the computational part into the chemical knowledge that I have, and also I work more on biology side here, and so that you have a much more what do you call like a holistic approach into drug design and development. So that's what you are going to see when I go ahead. So first I will give a brief idea about drug discovery. Then I will move into the various techniques that is available for carrying out computer-aided drug design with example from my lab, how we are applying the computational method to develop or design and develop molecule which show some kind of activity. Okay. And the third part will be, I will be specifically focusing on the de novo drug design part for designing some new molecule. I'll show some example again from my lab on. Okay. So what we'll do is like, once I finish, maybe I will talk for 1 hour and 1 hour 15 minutes. Then we can have some kind of questionnaire session. So I think I don't have to introduce what is a drug and those things, but two important aspects that we should always keep in mind when we are talking about drugs. One is we should always have a question whether it is a safe drug or not, whether that drug is effective for the disease we are talking about. So unless otherwise we prove these 2 points, we don't have a drug molecule going into the market for public consumption. So now we are having the problem of coronavirus, and people were talking about several drugs already. For the remdesivir is one example. Of course, now it is approved. At least in US, they had approved it for treating coronavirus. But still, there is a controversy going on, really, whether the remdesivir is effective or not. Because the WHO's clinical trial had shown that there's no advantage of using remdesivir, but still it is approved in US. Okay, so 2 aspects we have to always think about when we want to develop a new drug. So how these drugs work is first important thing that we want to know. So most of the drug work by interacting with a macromolecular target within our body. So when we are talking about a drug for diabetes, hypertension, or whatever it is, we are talking about a macromolecule or a biomolecule within our body. And when we are talking about a drug for coronavirus, then the drug target will be in that, either most of the time it will be in the virus. Okay, so drug target is important aspect. Like if you remember, like 2 sessions yesterday's, they were talking about the biomarker development or drug target identification using system biology approach, like a NGS analysis or the system biology approach to identify the drug targets. So whatever I'm going to talk about is not about drug target. So once you have a drug target, what happens? How we are going to develop a new drug molecule? That's what we are going to talk about. So many times when we talk about drug target, we are talking about proteins, specifically the enzymes we are talking about, because this is the largest class of drug target that we have approved drugs. Now let me come into this this specific example. I think all of us know what is this. This is paracetamol. Typically, I think every household, not every household, but many of the household will have some kind of stock in their inventory. Particularly if you have kids, I think this Crocin 120 will be there all the time, because you never know when the kids will get fever. So we will be keeping this stock in our house, and we all know this chemical structure. Because you are from pharmacy background, I don't have to talk about the paracetamol and the chemical structure. But anyhow, this is a paracetaminophenols. This is a chemical structure, and we have a phenolic OH group and a acetamide group with a benzene ring. So that I can go into the next part. So how it works is, paracetamol works by inhibiting the cyclooxygenase enzyme, so that it can reduce the inflammation. This is a major mechanism through which paracetamol works. But of course, there are other mechanism also people talk about for paracetamol, how it works. But most of these non-steroidal anti-inflammatory drugs that we are talking about, it inhibits the cyclooxygenase enzyme. So by interacting with this cyclooxygenase, paracetamol is able to inhibit this enzyme, so that prostaglandins are not produced, so that inflammation will be down. This is the molecular mechanism of how the paracetamol works. Now, if you ask someone whether we can make some new drug molecules based upon paracetamol, so that we can discover some new drugs. So if you ask a chemist or or pharmaceutical medicinal chemist, so by looking at this structure, immediately what will come into mind is, maybe we can make a homolog of this methyl. We can convert this methyl into a ethyl group, like CH2, CH3. This could be one option. Or we can, of course, we can increase the chain length. Another is, maybe we can change the amide into an ester function. That is another option that we can think about. Immediately it will come into your mind. If you're a chemist, you will immediately think what kind of modification you can do. And another can be, we can change the phenolic OH into an amino group, aniline, rather make it aniline. Or we can even change about the benzene ring into naphthyl, or maybe we can change into pyridine, pyrimidine, whatever. You can think about several other modification. A typical, what you call as analog synthesis. So if you are thinking about this kind of molecule, now the question immediately what has to come into mind is whether the molecule is safe, whether it is effective. Because these are the two primary criteria based on which a drug molecule gets approved in, or this criteria is essential for making a drug coming into the market. So immediately, if you think that, okay, you want to make this molecule, or you have this molecule, immediately what you have to do is, you have to make sure that it's safe and also effective. Now, how you will do this safety and effectiveness testing? There are two ways to go about. One is an in vitro assay testing. Another is in vivo. So in vitro assay, what we will do is, we will be testing this molecule, say example, in a test tube, or what you call this, in say 96 well plate, or in a laboratory condition outside the living organism. So what we need for this is, suppose you want to do this testing, you need, suppose we are talking about the paracetamol where cyclooxygenase is the enzyme. So we need the cyclooxygenase enzyme, and we need the molecule synthesized, so that we can do the in vitro testing to check either the effectiveness, or you want to check the toxicity, whether it is safe or not. Anyhow, we need the chemicals, we need the drug molecule, we need some kind of assay system for in vitro assay. Or if you are looking at the in vivo assay system, then you need animal animal models are required, you need the drug, and you can go about testing them. So both for carrying out the in vitro and in vivo assay, you need the physical presence of the concerned drug. Whatever assay system we are talking about, we need them physically present. Now there is an alternative to this in vitro and in vivo assay system to determine the effectiveness and the safety of this drug, that we call as in vivo testing, sorry, in silico testing, or what you call as generally computer-aided drug design. So this in silico methods, we can use computers to do the testing, both for effectiveness and for the toxicity. Here, you don't need the physical presence of the chemicals. You don't need the compound. You don't need the assay system to be set up. Everything can be virtually done. So that's what we are going to talk about. So I think I don't have to go through this typical drug discovery stage. So we have target identification, then we go for identifying lead molecule, then we'll go for preclinical, that is animal testing, then finally clinical testing, and go for marketing. This is a study. So typically your computer-aided drug design works from somewhere from hit identification, or what you call as a lead identification and lead optimization phase. So within this area only, your computer-aided drug design technique works. So again, going back to how we can identify a drug for a headache. So typically, so we were talking about the 3 drug molecule, newly designed molecule for the paracetamol analog. So what we have to do is, if we are going to really go about discovering a new molecule for treating diseases, you need to synthesize this molecule in the lab. So anyone knowledge with the chemistry background, they can go about synthesizing this molecule, or maybe similar molecule we can synthesize. That is one way you can, in the laboratory you can synthesize, or we can isolate some efficacy. So once this molecule, so if you have say 100 molecule, when you do the testing in vitro, what will happen is you may not find all of them effective. So from 100 molecule, you may find 10 molecules are good enough. So you take this 10 molecule and go into the next step. So a biologist or pharmacologist, we can do the use the develop animal model for whatever the disease we are talking about, and then we can do the testing of this 10 molecule to find out which is effective, which is safe. Okay, so based upon the preclinical investigation, you may find maybe 3 molecules out of the 10 what entered into the preclinical testing. You may find that they are good enough, means they are effective and safe in the animal model. You can go into testing in the clinics in humans. So out of the 3, you may find 1 molecule is good enough, means safe, and also it's effective, and eventually it will come into the market. Now the problem with this drug discovery setup is, people, when they analyzed many of the drugs which had come into the market, and what they found is, for every successful drug, there are almost like 5,000 compounds which had failed. I mean, due to several the in vitro stage or in vivo stage or in the clinical stage, they are found to be not promising, so they have been dropped. And the whole process of discovering a drug takes somewhere between 10 to 12 years or 15 years. So it depends upon, it may be less or more, but it takes a long duration and a lot of money is involved. So is there an alternative to avoid these failures or to cut short the time? That is the question. Now, this is where your CAD technique comes in. So there are several CAD techniques we are going to talk about. One of them is docking. So, so we can use such a CAD technique, and what it does is, when you have like a 3 or 10 molecule, when you design, you conceptualize that, okay, I want to synthesize this compound and go ahead testing. But what your computer-aided drug design technique is going to tell is, no, don't make all these 3 molecule. This out of these 3, only 1 molecule is promising. So remaining 2 may be a failure. So you don't have to work on this 2 molecule. You can avoid synthesizing these 2 molecules. Such a information it will give, or it will say you prioritize your effort. First, you test the molecule at the bottom, then only if you have enough resources and time, you can go to the other 2 molecules. So CAD technique is going to help you to avoid synthesizing all the molecule. So based upon the information from CAD technique, you can focus on a particular number of compound to synthesize. And of course, next you have to do the biological testing. You have to go for in vitro testing, in vivo testing, and finally you can test in human. And how this is going to help is it will minimize the amount of time and effort put in synthesizing many number of compounds. So this is going to reduce the time and also the cost that is involved in the drug discovery process. So this is the basic information, that is the advantage that we have. So now let me go into the second part. So we were talking about how it is going to be useful. Sorry, give me a second. Let's go into the various techniques that is available for computer-aided drug design methods. So you can classify the computer-aided drug design into 2 components. One is the structure-based drug design method that you typically we call as a direct method, and the second is indirect method you call as ligand-based drug design. So in the first method, we have a target structure known, and the second one, we don't have the target structure known. So when we use this, like, so you can see this quadrant. So if you know the target structure, particularly when you know the 3-dimensional structure of the target. So we are talking about cyclooxygenase. 3-dimensional structure is known for cyclooxygenase. And also you know ligand. When you have both this information, we normally use what we call as a docking method. So for docking, you should have 2 information. One is the protein structure, another is the ligand structure. Suppose you have only protein structure. Protein structure is known, or target structure is known, but you don't have any ligand. Then we apply what you call as a de novo drug design. It's very similar to your docking, but only thing is you don't have a ligand. But for carrying out docking, you need 2. One is the structure and is the protein structure and the ligand structure, both. And in case of de novo, we have only 3-dimensional structure of the protein, but ligand structure is not known. So we will talk about the de novo later. And if we don't have the target structure, due to some reason we don't have the structure, or we don't want to use the 3-dimensional structure of the target for designing new molecule, then we have ligand-based drug design. So in ligand-based drug design, we have what you call as a similarity-based searching can be applied, or more popular, we have Pharmaco4 based modeling and quantitative structure activity relationship. So these are the 3 techniques that we can apply when we don't have, or we don't want to use the 3-dimensional structure of the target protein. So Pharmaco4 searching and quantitative structure activity relationship. Suppose you don't have any information about the target, or we don't have any information about the ligand, then we have to rely completely on the experimental method to design new molecule. So either you need the protein structure or we need the ligand structure. These are minimum requirement for carrying out CAD techniques. When we don't have any information on this, then we cannot identify new molecules. We have to do completely experimental method. We have to rely on the experimental method to design new molecule or to find new molecules. Okay. So first we'll talk about what is docking. So simply put together, it's a computer algorithm that determines how a molecule bind in the active site of a protein. Because we know that drug work by interacting with a drug target to produce its effect. So suppose it's an enzyme, most of the time a drug molecule is an inhibitor, which will go on bind with the active site or substrate binding site of the target, and it will block, it will act as a competitive inhibitor, block the binding of a substrate, so that typically your enzyme is inactivated. It doesn't produce your product. It doesn't convert the substrate into product. That's how it works. So whatever is happening in the in vivo system, your docking is trying to simulate that process in silico, in the computer. So it's an automatic program that determines how a compound will bind in the active site of protein. And also it gives a score. That is more important. It will predict how is the binding, and also it gives some kind of a value. A numerical value is given. So the most important thing is for docking, like I said, you need the three-dimensional structure of the target, and you need the ligand molecule. Without this, we cannot go about doing a docking experiment. So this animation shows typically how the docking works. So what you can see is on the left side, you can see the protein target active site, and a small molecule is coming and binding into the active site. That's what you can see. I hope all of you can see this animation. So what is happening is you can see two things happening here in this animation. One, when the ligand is coming and binding into the active site, the ligand conformation is changing. Because mostly we are talking about small molecule drug, typically molecular weight less than 700 or something, 700 or 800. So and this small molecule will have a different conformation that it can take up. And another thing what you are observing is, like when the small molecule is binding into the active site of your target, you can see some conformational change is taking place in the side chains of the active site. So this is a typical example of what you call as a flexible docking, where both protein and ligand are considered flexible. Because if you in the in vivo system, when a protein and ligand, so we, if you some of you may be knowing that lock and key concept, where one key fits into one lock. That was the oldest theory. Now what we have is multiple locks, or multiple keys can open multiple locks, or what you call as a flexible docking or induced fit docking concept. So that's what we are seeing here. Both your target protein is flexible, and your small molecule is also able to take multiple conformation. This is your typical flexible docking. Okay, most of the docking program that we have, what we do is some kind of a semi-flexible docking, not a completely flexible docking or induced fit docking. Of course, we can run the induced fit docking also, where both can be both molecules, we can generate all possible conformation. But most of the time, the protein target, we keep it as a rigid, and only the small molecule will allow it to be flexible. Because making the target protein active site flexible, then your docking algorithm will take a lot of time to run, and you may, even for running one compound, you may take say 15 days or 1 month. That is not acceptable. So most of the docking program is like a semi-flexible docking, where protein is considered rigid and the small molecule only is considered as flexible. But we can, of course, run completely flexible docking. So what actually happens in the docking program? So basically the docking, there are two components in the docking software, whatever software we are talking about. The first component is what you call as a search algorithm. So what this search algorithm does is, it will try to generate all possible conformation, three-dimensional conformation of the ligand molecule that we submit. So for example, we are submitting a paracetamol. It will try to generate all possible conformation for the paracetamol, and it will try to fix these different poses in the active site of the target. So for example, it will generate, say, depending upon your input, it may generate, say, for example, 500 different poses for the paracetamol it will generate and put within the active site. Because normally the active site of target is much bigger than the molecule that we are talking about. Sorry. So the first component, that is the search algorithm, will develop or generate all possible conformations or poses, and then it will place it in the active site. Okay, so once these different poses of the ligand are placed, the second component, that is the scoring algorithm or scoring function, what it does is it will try to give a value, numerical value. So how this numerical value is given is depending upon what kind of interaction that particular pose is having with the active site. Whether there is a hydrogen bond interaction, whether there is an electrostatic interaction between the ligand, the particular ligand pose and the active site. So depending upon the active site residues present and the particular pose and what kind of functional groups are present in the small molecule, a specific score will be given to each and every pose. Suppose, like I told, like 500 poses are generated and put in the active site. So each and every pose, depending upon the interactions, it will be given a score. So this score is indirectly telling whether the particular pose is having better affinity to the target or not. This is your typical docking score. So docking score is going to tell about whether that particular pose is having better interaction with the target or not. So this is the same one. Pose of the molecule in the binding site is predicted, and the binding affinity or a score representing the strength of binding will be given by this algorithm. So this is the how this docking program works. So going back to what we need for carrying out a docking, we need a desktop computer. We need a software, because this docking has to be, we need some special software for carrying out the docking. One example is a Glide, which is a commercial software. Another is a Autodock, which is a freely accessible software. Autodock. And the third component we need, as I said, we need the three-dimensional structure of the target. So in this case, cyclooxygenase. We can retrieve it from protein data bank, which has a protein data bank ID 1PTH. I'll just come back to this protein data bank. And the fourth component we need is, we need the chemical structure of paracetamol. We can get it from different sources, or we can use some chemical drawing tools like a Marvin Sketch or ChemDraw or any other tool that you know about. We can draw the chemical structure. So these four components are essential for carrying out a docking. Actually, if you have a lab, if you have a hands-on lab, we can actually go about carrying out this docking in the lab. So we can download the from the protein data bank, like I said, the PDB ID. We can get the paracetamol structure from drug bank, and we can use the software Glide, Autodock, or any other, and we can generate the cyclooxygenase paracetamol complex structure. Okay. In silico using this software, and then we can go about analyzing this. So for the example that I talked about, that paracetamol, so I was talking about like whether we can synthesize 3 molecules. So what we did is actually I had done the docking of this analog 3 analog that we were talking about. We downloaded the PDB structure, and then we carried out using Glide. We had done the docking, and what kind of a docking score it gave. So paracetamol showed a docking score of minus 6.570 kilocalories per mole. So typically it's like based on energy values, binding energy is given. So you can see the minus 6.570. But if you see the other 3 molecules, you can see the one with the ethyl group is having minus 6.309, and ester is having minus 6.2 kilocalories per mole, and the aniline one is having minus 6.252. Now, based upon this analysis, what I can tell is none of these 3 compounds shows better affinity to the cyclooxygenase than paracetamol, because the more negative the score, more negative the docking score or the binding affinity, better is the molecule, or better it's interacting. The binding affinity is much stronger. So you will be looking for, when you look at when you are trying to analyze the docking result, you should have the docking score should be more negative. So here our control compound, the standard is a paracetamol. When we try to design new molecule, the new molecule should have better affinity to the target than paracetamol, or at least equal. Here, what I can say is from the docking result, I can say the 3 molecules that I have shown as an example, none of them are better than paracetamol in binding to the target. So I can, so if I have to proceed further, then I have to design new molecule. Maybe I have to, instead of the benzene, I have to go for pyrimidine or pyridine, or change to naphthalene or whatever it is. This molecule is not, these 3 molecules that I have shown is not suitable for actual synthesis and testing in the lab. That's what I can conclude from my docking study. So how this docking is useful? So you can see that it has predicted the activity of a drug, whether it is going to be active or not. And so by predicting that, it can identify new drug. So not only that, it helps to understand how a drug and target interacts. This is how the basically docking program can be helpful. So some of the tools where you can get the information regarding docking. Like I said, you need a 3-dimensional structure of target. So without that, you cannot go about carrying out docking. So one of the major place where people can get the 3-dimensional structure of a target is your protein data bank. So people use X-ray crystallography, NMR spectroscopy, or they use cryo-electron microscopy to solve the 3-dimensional structure of macromolecules. And those structures are deposited in this protein data bank. And this protein data bank is freely accessible by anyone. You can go there, whatever target you are, like in the case of cyclookinase, we can go ahead, search for cyclookinase, and we can download the PDB structure. There are other targets, related target informations are available in other databases. Another important is the therapeutic target database. So specific, so your protein data bank is going to there is information regarding different kind of macromolecules. So not only macromolecule in complex with ligands available there. Okay, so therapeutic database have information only about the therapeutic targets. You have super target, where around 6,000 drug target information is there. And so there are some situation where you don't have the 3-dimensional target information is not available with you. But you know what is the target, and what we can do is we can find the sequence of that particular target. So Uniprot is a place where you can find, if you're known, if it is a known protein, people are known about the protein, but we don't have the 3-dimensional structure. Like specifically, for example, many of the G protein coupled receptor, we don't have the 3-dimensional structure of that, because this is a membrane bound protein, and solving the membrane bound protein 3-dimensional structure is very difficult. So only very few we have in the protein data bank. So in such a case, what you will do? So you can get the sequence from the Uniprot database for the particular Gpcr. And there are some software, what you call as using homology modeling, we can, or using ab initio methods, we can construct the 3-dimensional structure of the target using modeler or ITESR servers are there. Swiss modeler is there. So there are, if 3-dimensional structure is not there, still we can go ahead, model the structures. So you have information about the target. Next is we need to have information about the ligand. So one important place where we can get the information is the drug bank. So all the approved drugs and many of the drug which is under advanced stages of clinical testing, information is available in drug bank. We can download the chemical structure of the particular drug we are talking about, or those drug which is coming into the advanced stages of clinical testing. We can download the chemical structure from the drug bank. Alternately, we can search PubChem. So I have given, this PPT that I'm giving a presentation, I will be sharing with Madam. I will also try to upload it into the Zoom chat box. You can get the information from there. So all the web links are provided here. So PubChem is the largest chemistry database from where we can retrieve the chemical structures of the compound that we are interested in. And Zinc is another important database why I am telling it's very important is, so this gives information about the chemical molecules which are purchasable. So if you are interested to, because eventually when we do the computational method, we have to go into testing. So when we have to go into testing, we need the physical presence of the molecule. So we need to purchase, or we have to synthesize. Two options we have. So if you use the zinc database for doing the docking, and when you find some kind of compound which is very promising, like for example, the 3 molecule, then when I did the docking, I showed -6 is the docking score. Suppose you have a compound which is having a -9 as the docking score, then we know that it's much better than paracetamol. So either you go and synthesize, or you try to search the zinc database to see whether we can purchase this compound from some chemical vendor. Okay, so we can always use zinc database. And another database, important database, is your Chembl. This is a European database. So what these people do is, they will screen all the medicinal chemistry related journals and find out all the chemical structure that they are being reported. Like for example, Journal of Medicinal Chemistry, European Journal of Medicinal Chemistry, several future medicinal chemistry, whatever journal, very top journals, they screen and find out what kind of compounds they are reporting, what is the biological activity. So all this information is stored in Chembl. So not only you have the chemical information, you have the biological activity. Suppose they are tested in, say, Gpcr, they are tested in, say, some enzymes. All the biological activity is also there. So Chembl is more useful for people who are working on ligand-based drug design, I will say. Like a QSAR modeling or Pharmaco4 modeling, it's very, because you have the ligand information and also you have the biological data information is available. Of course, there are many other databases. I just gave a few of the most important databases so people can access. And the third component that we need is your docking software. Without this, you cannot do the docking. So Autodock is a very popular open access software. One problem is that Autodock is you have to do multiple docking. For individual compound, you have to run the docking. So you have advanced version, what you call as Autodock Vina. Some of you may be aware, but many people know about Autodock, but not Autodock Vina. So this Autodock Vina, what you can use it for is, like, you can, like, suppose you have 100 molecule or 1000 molecule, you can do the docking of the 100 molecule at the same time to find out which molecule is having better docking. If you use the Autodock, each and every compound need to be docked individually. But when you use the Autodock Vina, you can have multiple compound, do the docking of multiple compound and get the result in one go. Or what you call as, we can use it for carrying out virtual screening. Okay, so that will come back later. Both are freely accessible. You have the link from where you can download and use. Another is UCF doc. So these are open access. There is a popular commercially available software, that is a Glide. Actually, our institute has a Glide with this is supplied by the Schrodinger group. So approximately 1 year license cost around 3.6 lakhs. We are trying to purchase, because we have the software, but it's going to expire, so we are trying to purchase. Another useful software is the Gold. This is also commercially available, but Gold is much more cheaper, one-time licensing. Glide is not like that. Every year you have to purchase a license. Another two software, one is the SwissDoc. This is again a free software. Another is a PatchDoc. Of course, there are, at least I can list out around 50 different open access softwares are available for carrying out docking. So this docking software, like, if you remember, I was talking about two components in docking. One is your post-prediction algorithm, another is the scoring function. So these softwares use different kind of algorithms, or the basic physics function that is used for scoring, they are completely different. Okay, so typically, whatever result you get in one docking software may not be exactly reproduced in another software. So don't try to compare. If you are trying to compare or reproduce somebody else's results, then you have to use exactly same experimental conditions, means in silico experimental condition, protocol you have to follow, use the same software. Then only you will get reproducible result. So this is one important thing you have to keep in mind. But many times I've seen Glide is a commercial software. It gives a very good accuracy because the scoring component uses is based upon experimental data. How they score it it depends upon the experimental data. But your Autodoc and Autodoc, we know Autodoc I think is based upon a physics-based function. We won't go into detail, but basic idea is there are some commercial software, there are some open access software. People who are interested, maybe you can start with the Autodoc Vina, a good place to start your docking. And if you have money, you can go about using Glide, but it's very expensive for academic, at least for academic purpose. Main thing you have to look into whether you can have sustainable. If you purchase for 1 year, once it expires, then after that we may not be able to use that software. So keep that in mind. But it's a very good, gives a very reproducible result. So to summarize this docking component, you need a 3-dimensional structure. You can get the 3-dimensional structure from PDB, or we can go about modeling the 3-dimensional structure. That is one part. Another part is you need a ligand for carrying out the docking, and you can get the ligand from zinc or PDB from the drug bank, or there are some virtual libraries available. That is the second component, and we need the software for carrying out the docking. Either we can use a commercially available or open access softwares. And once you carry out the docking, the results you can apply it for different purpose. Whether you can use it for binding mode analysis, or we can use it for virtual screening. We can use for structure-based lead optimization. We can use, or we can use for drug repurposing. So, drug.
Clear.
Madam, anything?
Okay, your voice is breaking, sir.
Am I audible now?
Yes, sir. Yes.
Yeah. Maybe some disturbance. So another useful thing is a drug repurposing. So drug repurposing is nothing but existing drug which is approved for a particular disease condition. We try to use it for a new disease condition. For example, this COVID, remdesivir is actually used for some other purpose, for Ebola and those things. So they try to repurpose it for coronavirus, COVID. Actually, for drug repurposing, docking is a very useful technique. Like I was talking about the drug bank, we have all the approved drugs that is there. What people have done is they had taken different targets for the COVID, the SARS-CoV-2 virus, and they had simply did docking of this drug bank compound. And I've seen more than 100 papers coming out in a very short time. People had done this docking and then say, okay, this molecule can be useful for docking, but without any experimental validation. But so actually, we have to eventually go for experimental validation to prove this molecule really that can work for COVID, but many times it's not there. But a lot of papers came for docking. People took use the Autodoc or any other software and did docking of this drug bank compound. But it can be really useful. And even in my lab also we had used, but I'm not going to talk about it. Another important application of your docking is what you call as a target identification or fishing, target fishing. Many times, like particularly when we are talking about natural product, we know they produce some kind of activity in say in vivo assay or in vitro assay, but exactly we cannot pinpoint what are the targets for that. So in such case, what we can do is we can say, for example, we're showing cancer activity. The natural product is able, like extract you have taken, and you find this is able to inhibit some cancer cell line, but you don't know exactly what is the target. What we can do is, like, you find out all the possible drug targets for cancer, and you can do the docking of this molecule on the target and find out which target is having better interaction. And based upon this docking information, we can go about and actually test the molecule for that particular target. This is we typically call as a target identification, target fishing experiment. Or docking can be used for fragment-based drug design also. So there are various applications in a different domain for docking. To tell the fact, out of the computer-aided drug design technique that we were talking about, the docking is the most popular and mostly used technique. There are several resources. Actually, recently I had completed editing a book by Elsevier. It's called Molecular Docking for Computer-Aided Drug Design. It specifically focused on the docking component. Of course, it's not yet published. It's in the production. Somewhere in 2021 March, it will be out. So there are several chapters in this book, complete molecular docking, basic, the theoretical aspect to the application, a step-by-step guide how to use, say, Autodoc, Vina software, and other software also is provided in this book. Maybe, of course, there are several other literature references are there. This one recent, it will be coming in next year. So that is about docking. Next part is the de novo drug design. So if you remember, when we have the protein information and the ligand information, we use the docking. When you have only the protein information, but we don't have the ligand. We don't have information about the ligand, and then we use de novo drug design. So here what happens is, actually, we know the target, a ligand actually is constructed in the active site of the target. So ligand molecule is constructed or designed from a scratch. So the older docking de novo program used to design molecule from atoms. You start with the carbon atom, nitrogen atom, oxygen. It will try to assemble a complete new molecule. But nowadays this concept is out. Now what the de novo program does is, it starts with a what you call as a fragments. Fragments are nothing but they are also ligand, but typically the fragment, the molecular weight is less than 250. Molecular weight is less than 250. And if you look at the Lipinski parameter, like the C log P is less than 3, the number of rotational bond is less than 3. So it's a very small, you can say it's not a complete molecule, it's a very small fragment. So we feed in a fragment to the de novo software, we give the protein structure, and it will try to construct the molecule ligand in the active site. This is what happens in your de novo drug design. So you need a target information, like what we need for docking, we need that information. Ligand information is not available, or you have carried out suppose you have carried out a docking experiment. For example, the cyclooxygenase experiment that we were talking about, the parasite, when I try to design some molecule and try to find its interaction, they are not at all good. So alternatively, what I can go is, I can go about doing a de novo drug design, because I have the target information. So what this de novo algorithm does is, first step is it analyzes the active site. So what it will analyze, it will try to find out what kind of active site residues are there, whether it's electrostatic side chains are there, whether it's a hydrophobic amino acids are there, like whether it's a phenylalanine is there, whether there is a glutamic acid is there, where it's placed. So this analysis it does. That is the step number 1. And the second step is, it builds a molecule in the active site. Depending upon what kind of interacting residues is there, it will try to assemble the ligand molecule within the active site in such a way that it will have better interaction with the active site. So basically, what you feed into the de novo program is the active the protein, and you will give ligand the fragments. So the building of molecule, and once the molecule is built, then it will be simply it's a docking experiment. The ligand is constructed, then it will do the docking of the ligand, and it will give a score, and you can rank order this molecule according to the docking score, and you can find out which molecule is having better interaction. So this building of the molecule in the active site, there are 2 ways to do it. One is a linking method, another is a growing method. So in linking method, what it does is, like I said, like first you will do the analysis of the active site. So there is an NH group there is a carbonyl group, or whatever it is. So the program, what it does, it tries to place the fragments. Like we say, there is a acetone group, there is a phenyl, there is a amine function. So these fragments are placed in the active site. So this placement will depend upon how is the active site, what kind of interacting groups are there. Once these fragments are placed, then using linkers, these fragments are linked to finally construct a new ligand in the active site. This is your linking method, how it works. So it will place the fragment. So in growing method, one fragment is placed at a time, and then it will be grown, depending upon what kind of functional group is in the active site. So basically, both the method you need. So depending upon the active site, the fragments are placed. Either they are placed at the same time and then linked, or it will be placed one fragment at a time and then it will be grown, or what you call as evolution. So after construct the whole molecule, then it will be simple docking process for finding out what is the score. So the main advantage of de novo method compared to the other method is the IPR, or what you call intellectual property rights. Because the molecule that is designed by de novo, typically they are not present in any of the database. So I was talking about the PubChem, which is the largest database. So most of the molecule that is known to us is present there. Somebody had synthesized, somebody had isolated. Drug bank, it will talk about all the drug molecule that is approved currently, which had withdrawn from the market, or which is going to be approved shortly. All the drug molecules are there. PubChem or Chembal. So somebody knows about this molecule. So only those things are there. So if you are going to take out those molecule and do the docking and go for experiment, eventually somebody could have done the same thing. Okay. So then you cannot go for filing a patent, because when you want to file a patent, you need to have a completely new molecule, because novelty is the primary criteria for getting patent. When you use the de novo method, we are taking some fragment and we are assembling. So many times what we found is the molecule that we design using the de novo method is not available in any of the database. So when it is not available in any of the database, nobody had synthesized this molecule, then we can get the proprietary right. We can go on file for a patent. That is the main advantage of a de novo method. Of course, it has a disadvantages. The difficulty, the disadvantage of your de novo method is the difficulty in synthesis. Sometimes, many times you will see that the molecule what is designed is difficult to synthesize in the lab, or it takes more steps. You may have to develop a new synthetic methodology to find this, because this is not already reported by somebody. So you need to synthesize the molecule by yourself. So that may be a problem. So there are again several softwares available in the domain. Ludi is one commercial available software for carrying out de novo method. So when I go into the de novo method, basically we use the Ludi. So this is a part of a Discovery Studio. Again, this is a commercial. We have a license for this one. We had used this. There are several open access software. One is a Lig Builder. This is you can download. This is a website is there. You can download and use. Another is a Lia 3D. Actually, this is a web server. We can submit the job there, and you can the de novo design molecule. It will give output. But Lig Builder is a standalone. You can download and use it. So the Lia 3D, we can use the FDA. So basically, it uses FDA approved drug derived fragments, because I said like we need fragments. So for this software, basically there are some inbuilt fragments available, or we can develop our own fragment and then we can give to these softwares. Because many times there are some vendors like a chemical, like a Sigma, Ulrich and all, where we can buy the chemical compound. There are some fragment libraries available with this software, with these companies. We can get those fragments and also use them for this kind of de novo experiments. So far we talked about structure-based drug design, where we need a minimum structural information. That is, you call as a structure-based drug design. So when we don't have the information about the structure, or when we don't want to use the structural information, we go about doing ligand-based drug design. So in that we have typically we have a Pharmaco4 modeling. So I think I have, it's almost 11 o'clock. I think I need to speed up. So Pharmaco4 modeling, typically we have a set of ligands. So at least somewhere around 10 to 20 ligand if we have, which show some kind of particular activity. So suppose you are tested using a cancer cell line and found is showing it's inhibiting a particular, say, HCT 160 or whatever, SW320 or whatever cell line, but we don't know exactly. It shows anti-cancer effect, but we don't know what is the target. So how you go about finding new molecules which can work similar to this? Because eventually for a drug to be approved, we need to know the target. Without knowing the target or exactly defining how the molecule is working, you cannot get the drug approved in the market. But before going, or we can do that simultaneously, but meantime, we can find out some new molecule. So for that, Pharmaco4 modeling is useful. So typically, when you say Pharmaco4, it's an abstract concept where you are trying to find out what is the three-dimensional arrangement of chemical or functional group which is present in those set of active molecule which is essential for activity. So typically, when you say a Pharmaco4 model, you will be talking about the where this hydrogen bond acceptor, donor, electronegative charge is placed in the space. What is the distance between them? So using this kind of Pharmaco4 model, we may be able to screen a library. Like what we do for a docking experiment, we can go ahead, screen the drug bank or zinc database for finding new molecules. Same type of thing using Pharmaco4 model. Once you design, you can use this Pharmaco4 model to screen these databases to identify some new molecule. Like I said, we can use it for modeling Gpcr Pharmaco4 model, because very difficult to get the three-dimensional structure of a target. We can use many times people use Pharmaco4 modeling to identify new molecule. Typically, it's useful for scaffold hopping or for patent busting. It's useful. Compared to docking, this is very fast. But the thing is, it's not like unlike your docking where you have a quantitative docking score. Like you can differentiate between two molecules, which one is better. Here we have a qualitative analysis. It can tell whether it will be active or not. It may not give a quantitative information. But this is a very fast technique, Pharmaco4 modeling. There are again several open source softwares and commercial. One is a DS Catalyst, is again from Discovery Studio. We have a license for this one. Actually, basically, we purchased this only this year. Another is Phase from Schrodinger. This is also both are commercial. There are Farmit, Zinc Farmer. These are open access. These are server based. We can submit the job and you can retrieve them. So they have inbuilt library. They'll screen and then they'll give the results. So people you can try to use this software. Another technique is QSR. Typically, QSR we apply when we have at least 30, 40 or more number of ligands are available. So typically, you can say this is like a mathematical equation. It derives a mathematical QSR or quantitative structure activity relationship. It's a mathematical relation between the biological activity and the molecular descriptors, or the physicochemical property or structural properties of the set of ligands. So set of ligand descriptors are derived from the chemical structures of the ligand, and you will try to correlate the descriptor with the biological activity using several statistical softwares. We won't go more deep into this. So using this QSAR, we can try to interpret how the molecule biological activity is there, or the pharmacokinetic property we try, or the toxicity. Typically, QSR is more used in the toxicity, determining the toxicity of molecule or how to avoid the toxicity of the molecules. So coming back to how to apply the CAD technique we talked about the basic CAD technique. So I will divide into two stages, how we can apply CAD technique, stage 1 and stage 2. Stage 1, I will say it's a virtual screening, or to identify a lead molecule for your project. So we have a big database that we talked about, zinc database, drug bank. So one option is you have the chemical molecule, you can go about doing in vitro testing, and then you can if you have the capability, you can test all the molecules and you can identify lead molecule. That is experimental method. But we can use virtual screening where we have the database, but we don't have to physically test all of them. What we do is use either the ligand-based method like a Pharmaco4 modeling, or we can use the docking-based method to test all the molecules in silico and choose the best molecules. Select, say, suppose you have 1,000 molecules, you can select only 10 molecules out of this database, and only this 10 molecule is tested in the in vitro. From that, you may identify lead molecules. This is your typical virtual screening. Either you can use the docking method, or you can use Pharmaco4 method or QSR-based methods. So depending upon the method, you can say structure-based virtual screening or ligand-based virtual screening. And eventually, you will be able to identify the molecule. So you don't have to spend a lot of time and money to test all the database. So one example from my lab. So basically, this is a work in collaboration with my postdoctoral boss from Taiwan, and a collaboration with the the chemical database we had obtained from the Taiwan. It has around 150,000 compound, and the assay system is set up by biotechnology department, Dr. Arun Kumar. So this is an epigenetic drug target called as G9A. It's known for cancer. So instead of testing all the molecule, what we did is from the literature we identified this is many of the drugs that is reported. It has this specific pyrimidine structure. So what we did is we took this structure and we queried this database, what you call as a substructure searching. When we use the substructure searching, we got around 300 molecules we had taken out. So this 300 molecule, what we did, again, it's not possible to test all the molecule because we have to take from Taiwan. So we did a docking-based virtual screening for the 300 compound, and we identified potential 6 molecules. So this 6 molecule, we asked our collaborator to send from Taiwan. And when they send this 6 molecule, and the biotechnology department, they tested this molecule, and we identify one molecule, CSV, some number. This molecule shows it can inhibit the G9A enzyme, and also it can actually go inside the cell and also inhibit the methylation. Because G9A is it methylates the histone methylating enzyme. Basically, it can inhibit the cell. You can see the different concentration. You can see the methylated histone. You can see the level is going down. So this is a typical. So instead of testing all the molecule, we had tested 6 molecule, and we had identified lead molecule for the particular target. So what are the factors that consider? You need to understand the biology or structural biology of the target. Because this is published in Biochem. Actually, before this, we published one more paper because there we analyze the active site actually, and we find some specific residues where the interaction should be there. Okay, so because the active site is very big, exactly when we did the docking, the molecule actually binds in different places. So not all the interactions are productive. So basically, we need to analyze this active site where, because to tell the fact, if you take a target and you do the docking, most of the time there will be some kind of hits will be available. But really, they're going to be going to be effective or not, that we have to find out. Okay, so for that, we need to really understand the structural biology, and you need to find some way to exactly find out which are the residues, key interacting residues. Okay, that we need to find from the target information, and whenever we know some information about the known ligands, this is going to be helpful. When we have when we are trying to find a molecule for which there is no known molecule for a target, then it's really your docking experiment is going to be very tricky. You cannot blindly use docking to go ahead and test molecule. If you know some ligand molecule which is really interacting with the target, then we have to use that information to find new molecules. It's going to be very helpful. And the second stage is so first you have identified lead molecule. Next is you have to go for what you call as a lead optimization. So this is your second stage. So let's see how we can use CAD techniques. So we know why we need to do the lead optimization, because the molecule may not have the lead molecule may not or whatever the hit you had identified is not optimal. This interaction is not optimal. This toxic, maybe ADME property is not good. Whatever it is, reason we can typically for lead optimization we use medicinal chemistry optimization, where you do change the chemical functional group. Like for example, that paracetamol I was talking about, you can change the amide to ester. We can change. You can increase the chain length. You can convert phenolic OH into NH2. This is a typical medicinal chemistry optimization. So this one based upon a knowledge based, we are trying to do that, but it may take more time. This where your CAD computer aided drug design comes into play. So once you know how the protein ligand interacts, based upon the interaction, you can find where to do the modification, what kind of modification you can do. Such an insight can be given from your CAD method. So using this, you can synthesize new molecule based upon the CAD, and then you can go ahead, do the in vitro testing. And if you find better molecule, you can move ahead, or you can go back and do the further lead optimization. So this is an iterative process. It will take several rounds. And when we use the CAD method, this will be very helpful. It will minimize the number of molecules that we actually synthesize and test. So this is an example from my lab, how we use the lead optimization method using computer-aided method. Madam, how much time I have, madam?
Sir, no problem. You can take, sir.
Okay, okay. So maybe I'll try to finish in next 20 minutes. Basically, this is a project from a DBT Northeast Training Program. I got working with Dr. Suresh Kumar from NERIST, Nirjuli. Actually, what he wanted to do is like this is a plant, Cleodendrum colubricum. This is a traditional medicinal plant used for hypertension. Actually, what the collaborator, what he was telling is this is normally available in the market. People consume this like a vegetable, and this is known to have a hypertension property. So what we did is we want to know why we want to use computational approaches and find out whether what kind of target on those things. So when we did the literature search, what we found is people had extracted this plant and they reported that it can have interaction with different target. Like one is a ROC target, which is antihypertensive target. Another is a ACE inhibitor. They showed. So some people are saying it's having ACE inhibition. Some is telling ROC inhibition. Some was telling PDE inhibition. So what we did is we used 3 different target, and in literature there are 21 different constituents reported for this plant. We took out all this and we did the docking, and what we found is this particular plant astoside is having better interaction with the ROC. Of course, there are other other compound also we found, but this is more promising. And so simultaneously, what we did is for the ROC, there is one approved drug called Fasudil. This is approved for not in India. It's approved in Japan. So when we did the docking for this compound also, what we because this is a crystal structure is available for this Fasudil, and when we did this, what we found is this isoquinoline ring and this what do you call as this caffeic acid, the phenolic component, these are binding at the same place. So not only we did the docking, we did further extension like with molecular dynamic simulation, and we found this. This is the key. The caffeic acid component is the key interacting residue with the ROC, because ROC is a kinase enzyme. Typically, we need a stable interaction with the methionine 157, which is a hinge region residue. We call this. This interaction is a hinge residue interaction is a key in all the kinases. And here, ROC, this is methionine 157. So we found this. And another important thing, what we found is the homopyperazine ring that is present in the Fasudil is placed somewhere in the place where the sugar moieties are available. And another thing, what we know about acetoside is this is a glycoside, and typically it's very cumbersome to develop the glycoside as a drug molecule. So another important thing what we want to do is we want to get rid of the glycoside. So what we designed is whether we can replace this homopyperazine ring, or we can make a hybrid design with a homopyperazine ring attached to this caffeic acid. This is a basic. So we designed a new molecule, and we did several docking and MD simulation. Another important concept is you can see the rule of 5, that is Lipinski rule. You have 3 violations for acetoside, and when the newly designed molecule, so what it means is the pharmacokinetics will be better for the newly designed molecule. And we designed the molecule, and we did the docking, molecular dynamics simulation, all those things, and we found this really interesting molecule. Actually, we tested this molecule and in cancer cell line, because ROC is not only a target for hypertension, it's also a good target for anti-metastatic to prevent the invasion. So we could in our lab we can test this molecule. We got it synthesized from Vellore Institute of Technology from the chemistry department, and we tested this molecule. It showed anti-cancer effect in colon cancer cell line. But one important thing what we can observe here, this is a carbamate, because basically this is intermediate when we synthesize the molecule, hybrid molecule, and this intermediate showed better activity than the actual molecule. So when we did the docking, we saw that this particular functional carbamate group was making specific interaction with arginine residue in the active site. So what we thought is this carbamate being not very stable, we designed urea and thiourea to replace this molecule. So urea and thiourea will be much more metabolically stable. So we designed this molecule, and we carried out a docking study, molecular dynamic simulation, all those things. And eventually when we tested, we found this molecule is showing a better anti-cancer profile than the your phosphodil. So we had used a integrated computational approach along with the synthetic and the biological experimentation to identify lead molecule and also to optimize the lead molecule. Of course, further design to improve, because now only it's in the in vitro stage. This is a thiourea. We can take it further for much more intensive testing of this molecule. So what we need, what are the factors that we need to consider is what is the target ligand lead molecule binding orientation or interaction. At the same time, we should have some basic information about the organic chemistry or the synthesis we should understand. Without such a knowledge, we cannot design, like blindly we cannot design the molecule based upon the computer input, because the computer is not going to tell what kind of molecule you are going to design. And when you're designing it, at the same time, we should have some idea about how to make this molecule also. And of course, you have a reproducible bioassay is required. Without having a reproducible bioassay, it's very difficult to go about making molecules. And let me move into the case study that I was talking about for the de novo. So this is basically a continuation of the lead optimization that I was talking about. So because this is my PhD students' work, one direction we are going based upon the structure-based drug design concept. And alternately, what we did is we want to use the de novo method. Like I said, de novo method, we are trying to construct a ligand molecule in the active site using fragments. So here we have, so what fragment we identified is caffeic acid is a very good fragment which will interact with the kinase, the hinge region residue, because when we use this fragment and we did the molecular dynamics, it was making a very stable interaction. It was not coming out, because typically when it's a very small molecule, it may move around here and there to do the, it can interact with many residue, but that was not the case. This caffeic acid is specifically interacting with the methionine residue in the active site. So we thought maybe this is a very good fragment to start our de novo design. So we use this fragment as our base fragment to construct the new ligand molecules. So this is one fragment, and we need multiple fragments for constructing the de novo molecule. So what we did is, from literature, we found 372 known ROC inhibitors we identified. And we took out all this, because these are known ligands for the target. We took this and we used a program called as mole blocks. So what this mole block does is it chops up the ligand. If you give a drug, a molecule, it will break down into smaller fragments. So how it fragments is, it uses what you call as a recap algorithm or retrosynthetic combinator analysis procedure it uses. So if there is an amide bond in the ligand that you give, it will break the amide bond into amine and a acid fragment. If you give a ester, it will break it into an alcohol and an acid fragment. So whatever ligand you are giving, it will break according to this 11, basically 11 different reaction types are there. Based upon this, it will break it down. So what actually basically why we are trying to break according to the reaction is you can assemble back this fragment into a ligand molecule and it will be easy. Suppose if you break an amide bond into an acid and amine, you can use the acid amine coupling reaction to bring back the same molecule. Okay, so using this recap algorithm, this mole block will fragment it into different fragments. So when we fed 372 ROCK inhibitor, it had generated 447 fragments, unique fragments. So why we use these inhibitors is because already these inhibitors are able to interact with the target. So we want to find some fragments which will be binding, it has a better chance of binding with the active site. So we use this known inhibitor, we generated 427 fragments, and using the Ludi algorithm, MCS docking, multiple simultaneous docking, it had generated different poses for this fragment. So basically this algorithm, what it does is it will try to find out where these fragments will be placed in the active site. Like if you remember, I had shown in the previous in the de novo, we need to place the fragments in different site and then you need to connect. So this MCS docking, we put this different fragment in the active site, and then we go about the de novo evolution. That is the growing method, and it had designed and it had given output. So based upon the output, we had designed few new molecule and then go about identify some new molecule, and I'll show you the results. Okay, so this is the MCS docking result. So what it does, so basically we have the base fragment, that is the caffeic acid, because we know that this is the critical interacting residue. So when we gave the 470 fragment, it had identified 5 different fragments, 1, 2, 3, 4, 5, and it had found that the particular where is the possible linking position. So this 5 fragment is the one which is having better interaction with this, and it gives us some kind of score, MCS scoring. So you can see all of them, it try to give a linkage with the methyl group. So this could be a important point for linkage with the carboxylic group of your caffeic acid. So what we, based upon the score, okay, let me see. So this is one fragment and the other fragment is here. So it had given M120 and M49. So basically, based upon the input from the docking algorithm, we had designed this molecule through the ester linkage, and this is caffeic acid M49 ester, and this is 120. Based on that, we have designed another molecule. But we know that ester are not highly stable, metabolically not very stable. Of course, this is a phenolic ester. We cannot just rule out this molecule. But what we thought is we will use both the phenolic ester and also the amide. This is based upon our medicinal chemistry knowledge that amides are much more stable than the esters. So basically, we designed 4 molecules, and all the 4 molecules were docked. And what we found is the caffeic acid amide 7A is having better docking score in the active site. This is the 7A molecule. And another important thing is we focus only on this molecule further because if you see the other molecule M120, there is a chiral center. Again, there will be problematic for synthesis. So we don't want to go into that part. So we just kept this one. And another thing, when we see a free amine as a medicinal chemist, immediately what will come into mind is we designed the amide, urea, and the sulfonamide. And all these molecules, we can see the Lipinski rule is like within the limits. And we docked all the amide and those things. Another important thing what we could find is, sorry, when we analyze the crystal structure, we saw that some of the known inhibitors are binding with specifically in the cavity A and cavity B. This we found out when we analyzed around 11 crystal structure. And some of the newly designed molecule you can see specifically is making interaction with cavity A. Some in, like this is making with cavity B, 2 molecule, 7A, 8A, but 9A and 10, the sulfonamide and the urea is making interaction with cavity A. And docking score, you can see 10A is having a good docking score. Of course, 7A is having better docking score. And we did molecular dynamic simulation and several other computational methods. And clearly, it shows that the most key interacting residue is methionine 156. It's making stable interaction with this, because without this interaction, it's not worth synthesizing this molecule and testing. But we can see this. Actually, I'm not showing the phosphodil data. It's also making similar kind of, it's like, I think it's a 20 nanosecond simulation data. The hydrogen bond is intact. Means it's not. So molecular dynamics is, so molecular docking is like a statically you're seeing whether there's an interaction or not. But in molecular dynamics simulation, what we do is in an aqueous environment, we try to simulate the molecule and see whether the interaction is stable. So this is more like a dynamic experiment. If the molecule is not having strong interaction, the ligand actually dissociates from the target. Many times I have seen this, at least sometimes I have seen if it's a not strongly interacting residue ligand, it will come out of the active site. But that we are not seeing here. So we can go about synthesize and test this molecule. This is a molecular dynamic simulation. So overall, if I say we have started with a plant constituent, one direction we have tried to use the structure-based drug design method to identify some new lead molecule. We had synthesized and tested. And simultaneously, we have designed this some new molecule. So whatever the de novo design molecule, we have searched in Zinc database and the PubChem, and exactly these molecules are not available. Very closely related structures are available, but these are not available. So we can synthesize then test it, and there is a chance that we can get a patent. But we are not gone into that stage due to limitation. So there is case study 1 and case studies 2. Typically, so this is more like a development of an algorithm for de novo drug design. This is in collaboration with the computer science department, Professor Sivasatya. Actually, she is going to give a talk in the afternoon. So basically, because computer scientists, they try to develop algorithms. So she's an expert on multi-optimization parameter optimization techniques. And so we collaborated, I collaborated with her to design some new algorithm for de novo drug design. So I won't go into that. So basically the concept is, again, so you have multiple fragments and you try to assemble those fragments. So whatever I was talking about is the structure base. You need the protein structure. So based upon the active site, we are trying to design the new molecule. But in this de novo, whatever we have done, it's not based upon the active site. Instead of that, we need a template chemical structure. A lead molecule is required. So you, for example, if you have a paracetamol, based on the similarity concept, we can design a new molecule. So sometimes this kind of de novo drug design is also used. More popular is structure-based, and ligand-based de novo is also there. So we use this concept. So once you design the molecule, of course, we have to go ahead, evaluate the molecule and test it. So for this, basically, so Bioinspired Algorithm uses the concept of biological, every organism has some kind of special features. So here, monkeys, they have some kind of climb somersault behavior. So they developed a new algorithm. So one important concept is, so here what we do is, you generate a molecule. So each molecule will be generated from different fragments. So here we use the acid fragment, amine fragment, and diamines. So you can see there is an acid fragment is there, and amine and a diamine fragment. So these fragments are combined to produce the final molecule. So so initially how the algorithm works is it will initiate, it will generate a population of monkeys. So that is representing basically a molecule. Okay, so when you say this is the position in 3 dimensions, so basically the molecule is, the chemical structure is this one. So you have an acid and then you have an amine. So when this program runs, it need to evaluate whether this molecule, what is its fitness. So the fitness function here is similarity to the known drug, that is a lidocaine. So we had used the lidocaine. We want to design molecule which is similar to lidocaine. Another is another anti-cancer drug. So we can take any kind of drug molecule. We will try to design molecule very similar to that, but it is an unknown molecule. That is the concept of our de novo. So we have taken lidocaine. This is a standard. So for the fitness function, one is Tanimoto similarity, and another is Lipinski rule of 5. Whether this molecule will obey the Lipinski rule of say C log P or your molecular weight. So these are the criteria for evaluating the molecule. So once the algorithm runs, it will generate a lot of molecules and it will try to evaluate. So for example, this furanopyrimidine, typically it doesn't obey the Lipinski rule because it's too big a molecule. But when we design the molecule, when the algorithm runs and then terminates, we identify several molecules which is like obeys the Lipinski rule. So you can see the lidocaine, based on that, it has generated molecule with oral bioavailability score is 1, means it is obeying all the Lipinski rule. And the Tanimoto similarity, you can see 0.5752. For this one, it has generated several molecules. You can see some of the molecules that is generated is like the OBS score is not good enough, but the Lipinski, the Tanimoto similarity is somewhere around 0.53. So this new molecule, so when we search this molecule in Zinc database, they are not available. Only closely related molecule is there, because this is a de novo method. We use a different fragment to construct, and these are not reported. So this we had published in International Journal of Intelligent System and Application. Actually, we had developed other algorithm also. The computer science department, they developed what do you call as biofilm algorithm. This was so this we had used specifically for designing new anti-diabetic drug based upon natural products. This is published in Applied Soft Computing. This is a very good journal. So with this, I had basically I wind up my talk. I didn't touch upon the artificial intelligence because this is not my expertise. I don't use artificial intelligence into computer-aided drug design method. But people, I think they are going to talk about later. I just put one recent paper in Chemical Reviews. It's a very good article. Another, there's a special issue in Journal of Medicinal Chemistry which talks about various artificial intelligence techniques in drug design. People interested can follow this. So for applying, for developing new drug, I think you need a very good collaborative team between chemist, biotechnist, biochemist, and a computational scientist to do drug design development. So I would like to thank my different funding agencies. So the case study, one is from Dr. Hemant Arya, case study 2 from computer science, Dr. Vasundhara Devi, and my collaborators, Taiwan, my postdoctoral boss, and Dr. Suresh Kumar from NERIST, Dr. Kannadasan from BIT, and Dr. Arun Kumar from Biotechnology, Pondicherry, and Professor Sivasatya from Computer Science PU. I'd like to thank them. Thank you. I'll take any questions I can take.
Thank you very much, sir. Pooja? Pooja, are you there? So some questions are posed by the participants.
Okay, madam.
The first question is, 1 minute. When cinnamic acid synthetic example is attaching to same place, which will bind to the site in the newly designed
exactly
molecule?
same the phenolic OH exactly in all the molecule that we had. Actually, I'm just showing only the caffeic acid, the fragment. Okay, actually basically we got 3 molecules. One is ferulic acid, caffeic acid, and para-coumaric acid. All of them has a OH group. So in all the 3 molecules, we are seeing the same kind of interaction. It's exactly because that kinase, that hinge region interaction is very, very important. So everywhere we are seeing that interaction is the same, because when you attach new group, no, it should not change the orientation. That never happened. And in all the docking, we had used glide. It gives very reliable result. If you use autodock, maybe it will give different kind of result. And orientation, if the orientation changes, then there is a big confusion, but it never happened.
Sir, which software is best, Autodoc or any other freely available? Not you should not compare with the Schrodinger.
Yeah. So I think in that case, I think Autodoc Vina is pretty good. But I will suggest that when you do the docking, like I given like 4 or 5 list, you can use 2 or 3 software to confirm your result.
Okay. Some of them are using Swiss Doc, sir. What about
yes,
that?
That's what. You can use Swiss Doc, iGem Doc. Several softwares are there. At least 2 software you try to use.
Okay. Swiss
Doc. And particularly when you're going to do only the docking and report, if you use the 2 software and if you can produce reproducible result, similar result, then there is a high chance that your paper will get accepted instead of just.
Is it possible, sir, by using the 2 different softwares, 2 different softwares with the same molecule, with the same will show the same type of binding energy?
That's what. If you are really your molecule is going to be really good enough, your predictions are really good. If really there is going to be activity, interaction, then it will give the same result.
Okay.
Then only, it's like a confirmatory.
Okay. So there more than 2 or more than 1 software we have to use when we are going for free.
Yeah. At least 2. Even like I had used, actually we have a Discovery Studio docking software. For some other work we have done, when we do the docking, it doesn't give the same result. Glide and Discovery Studio, but when we tested, some molecule from Discovery Studio was active, some molecule from Glide it had been active. So it's not guaranteed that when you use a Glide you will get always active compound. Okay. So because again, virtual screening means it's computational. I will say maybe 10% you will get real result.
Okay.
Of course, it depends upon what kind of target you are using.
Somebody asked that, is it related to free energy docking score?
Yes. Binding free energy. Yeah.
And the next, sir, please do discuss about the how modeling of GPCR can be done. Is there any software?
No, you have to. So that's what your structure. You have to. There is a modeler. Okay, modeler uses a. So GPCR already some crystal structures are available.
Oh.
Okay, so we can take that crystal structure, use as a template, and you can do homology modeling.
Oh.
Okay, because I didn't touch upon those things. So homology modeling, it needs already a template. Because there are some GPCR, we can use that to model it. Or we can use completely ab initio method, like what you call as ITESR. There is a server, you can submit your protein sequence. Once you submit it, you need to give the email ID, and it will say it will take 2 or 3 days, it will give you the structures. And there are ways to validate those structure, model structure. Okay, I didn't go about into those things, but there are a lot of literature related with the modeler. Swiss PDB also, that also you can use it for modeling.
So is there any software to predict or to find out the active site, how to pick the suitable PDB?
So, okay, so when we do the docking, we need to know where is the active site. We cannot just do the docking in the. So some of the software you use a blind docking, so you don't have to define the active site. But many times you need to define the active site. So active site will depend when you take the PDB, if there is a known ligand, then we know where is the active site. Alternately, there are some softwares like online tools, like CAST-P is one popular tool. When you feed that PDB ID, it will tell what are the active site residues. Okay. Using that information, we can give it to Glide or to Autodoc to do the docking.
Okay. So plenty of questions are there, sir. So once we have identified the target and ligand, how long will it take to synthesize a drug? After synthesis, how long it will take to market?
So this is a basic question. I think that's what I showed, because being a pharmacy people, I just skip this thing. It takes like about 10 to 10 years from the initial project starting to your marketing, it will take
Marketing.
yeah
In
10 years.
conventional method,
Yeah,
in
conventional.
CAD,
Yeah.
when you go for CAD, how
Maybe
much time they are
okay,
asking?
okay, that okay. So exactly we cannot quantify.
Okay.
So people say like if you see the process I given, how I start with a small what do you call, from the plant product, how I come up to small the lead optimize some of the compound which is showing activity. When we don't have any of this computation method, we don't know whether the glucose part is interacting, or whether the phenolic, whether the which part is interacting with the key residue, that we don't have any information.
Okay.
Without that computational docking, there is no way. I cannot tell which part I have to do, whether I have to retain the glycoside part, or I have to retain the phenolic part, whether the benzene ring I have to retain, whether the double bond is important, I don't know.
Yes.
Okay, so that kind of insight only it can give. It's not going to tell exactly, okay, you can tell only 100 molecule, you don't have to synthesize, but 5 molecule. But exactly what molecule actually I designed is how I brought in the homopiperazine ring is based upon typical medicinal chemistry knowledge from the literature.
Okay.
Okay,
Sir.
the computer didn't tell me I have
Yes,
to put
sir.
that homopiperazine.
Sir, how two ligands can be docked at the same time, sir?
One by one we can do that. That's what multiple simultaneous docking that I was talking about in the de novo. So multiple molecule can be docked into that.
Sir, last question, sir. So many are asked, but I ask because of time constraints, I'm asking, what is the difference between the docking energy and binding energy? If they are same, why different terminology?
Yes, actually, the algorithm, what do you call? Typically, this molecular mechanics force field is used for determining the docking score or binding energy. So they will use different, whether there will be some kind of penalty is used or not. Because sometimes, like when the molecule is binding, they will have like a different. Because I told that the post generation it will generate a lot of poses, right, in the active site. So some of the posts may not be at the minimum energy level.
Okay.
So due to that, the docking score typically will use some kind of penalty. Okay, and solvation energy, all those things will be computed. But binding energy, it may not be using those. So, so because of that, there will be difference between the binding energy and the docking score. Okay, so you should be careful in looking at this part. Like, so every software will tell what exactly being used for the docking score or for the binding energy. They may not be same. Sometimes they may be same, depending upon what are the components. That you have to carefully look into that.
Okay. There are, sir, but
How MOP? What is this MOP? Is multi-objective optimization parameter technology, or what is it?
yes,
So that's what I was talking about in the de novo method, am I right? So typically, like when we talk about a drug design, you have to optimize multiple parameters. Like in the de novo, what we were trying to optimize, one is the Tanimoto similarity, another is your Lipinski rule, like the molecular weight, another is a C log P, hydrogen bond acceptor, donor. So these are multiple parameters we have to optimize. So so if, for example, the second example where I talked about an anti-cancer drug, the molecular weight is very high. So we have to find a molecule which will be similar to that. At the same time, the Lipinski rule also should obey. So when the algorithm is running, it will try to find a molecule which is the Tanimoto similarity should be also near 1. At the same time, the other parameters also should be nearing 1. But we cannot get a molecule which is like always 1, because that is also not good. It will be same molecule. So depending upon the fragment, it will try to find which is the most optimal one.
Okay. Okay, sir. Thank you.
Thank you very much, madam.
Sir,
I hope it is useful.
Sir. And
And
very much, sir. You have started your talk with the very basic things which may be very easily understand to even layman also. You have started with the drug and how the drug works and how it is evaluated and what are the drug discovery stages and identification of new drug. And not only that, you are also compared your things, conventional method with an CAD and how to cut short the time and the money by using of this in silico studies, and then you move to an CAD and the components of the various components of the docking softwares and requirements of the docking and how to predict the docking results and what are the uses of docking and tools for the docking. And you are also touched the drug repurposing concept and how this drug repurposing concept is useful in silico studies and why and where we can use this drug design and how to work it and advantages and disadvantages of de novo methods. And you are also touched the QSR and with a better examples you are explained the lead identification, optimization, de novo design and evaluation, and how the monkey algorithm and biofilm algorithm are used in the de novo design. Sir, your session is very, very elaborative, informative, and thought-provoking. I think most of the participants will start doing the research in this field because it is a very informative and interesting field, sir. Thank you very much. On my own behalf and the organizer's behalf, I express my heartfelt thanks to you, sir. Thank you very much.
Thank you very much, madam. I just uploaded that file, that PPT. I just uploaded so everyone can use it. I hope.
Thank you, sir.
Thank you very much, madam.
Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Yeah.
Dear participants, you may have a 5 minutes break, and after 5 minutes we will start our session. Okay.
Shall I start, madam?
Slide now. Pooja has to share the slide.
Pooja, can you please share the slide? Pooja, are you there?
She's there. Pooja?
Good morning, dear participants. Welcome once again to the second session on second day. For this session, I propose hearty welcome to Dr. Girinath Pillai. Sir, are you available?
Sir.
Girinath, sir?
Pillai, sir. Actually, sir said that he is in.
Sir is unable to unmute, madam. So you have to unmute, sir. Sir is available, but he's unable to unmute.
So I had to do, na?
Yes, ma'am. You have to do and.
Okay. 1 minute.
Good morning, everyone. Yes, madam. I hope you can hear me now.
Okay.
Yes, sir. Yes, sir.
Thank you. Yeah.
So on behalf of Institute of Pharmaceutical Technology, on my own behalf, I'm very happy, glad to welcome you to the second session of the Faculty Development Program organized by IPT with the support of AICT. Thank you, sir, for accepting our invitation and be part of this FDP program. To give a brief introduction about biodata of Dr. Girinath G. Pillai, who is currently working as Chief Scientific Officer, Jastra Innovations Private Limited, Bengaluru. Before joining this Jastra Innovation, sir has a wide research experience and working experience as lecturer, scientific support manager, technical consultant, etc. And sir has a very strong research background, which is from his academics. Sir obtained MSc Bioinformatics from Bharathidasan University, MPhil Biotechnology from Periyar University, and did his Doctor of Philosophy from University of Tartu and University of Florida. Sir was once awardee of a prestigious Marie Curie Actions Research Fellow. Sir also worked as project fellow and RTA research specialist in University of Tartu, Estonia. Now, currently, sir is not only Chief Scientific Officer in Jastra Innovations, but also working as Technical Consultant India Material Design, Research Advisor in Vellore Institute of Technology, Scientific Advisor for Nairo Research, Chief Scientific Officer of Jastra Innovations, as I already mentioned. But most importantly, sir also is serving as a mentor and program coordinator for Drug Discovery Hackathon conducted by MHRD Innovation Cell India recently. Sir has a vast experience of 14 years in computational chemistry, molecular modeling, and QSR. And sir is a regular resource person in more than 72 training workshops where he is dedicated to train the student and faculty with various computer-aided drug designing softwares. Sir mainly has conducted workshops in the area for Biova Accelerators, Discovery Studio, FlexX, Turbomol, Cosmotherm, and Record softwares. Now I invite sir to give his talk on machine learning in drug discovery and drug candidate selection. Welcome you, sir, once again. And now over to you, sir.
Thank you so much. Let me share my screen. I hope you can see my screen now.
Yes, sir.
Great. Okay, so I will start then. So thank you all. Thank you so much for the nice introduction. And I also extend my thanks to Dr. Sridevi, Madam, who has given me an opportunity to talk about this. And now, and as per the introduction says, I love training and transfer of knowledge. That is my key motto for the next coming years. Being mentor to many, and I'm also happy to be part of this as an Atal sponsored from IICT, because I'm also one of the mentor of change for Niti Aayog's Atal Innovation Labs for schools. So we are promoting research in school students too. So 3 schools are being assigned to me and mentoring them. So like that, I'm happy to be here. I hope many of the majority of the audience are students. I mean, the participants are students, and I could see many more faculties and known faces there. So today I'll be talking about machine learning in drug discovery for drug candidate selection. Many things are bit and pieces put into this particular title. And I have to start with a disclaimer anyway. But what is this drug candidate selection? We have to see how many drugs came into the market in India or from Indian academics. If you see many of the pharma drugs that is marketed, many of them have a good history of coming from academic research, either patented from academics and getting transformed and commercialized. That's what we can see many of the histories. But in India, why don't we see that much? If it is there, please correct me. I might not be updated. I'm talking about FDA molecules, small molecules, or new molecular entities or new chemical entities. I'm not talking about nutraceuticals or herbals or anything like that. Of course, there are plenty of them anyway, and up to a credit as of from India. So that's where we are looking at how academics can help to reach to a drug candidate. Rest of the part will be taken care by the regulatory affairs and people who have to take care of it. So how we can do that? In order to do that, the important thing is that we have to avoid failures. As Professor Mohan already mentioned, it is not that using a software you can find a drug. Not at all possible. There could be a good synergies between human expertise and computational tools. Computational tools are allowing you to generate a lot of data. And you are the master to consider whether that data to be validated, considered or discarded, or how much to be considered. So you need to have that much of expertise. So don't consider any tools or softwares as a click a button, getting the numbers and reporting the numbers is not a research. May be useful for your publications. But if you want to transform your information or knowledge from the desk or what bench work you're doing to a scalable marketing solution, that's where we have to see signs of relevance. So that's where I am more focusing today with this presentation. And let me start with this disclaimer. We are not yet completely ready with AI or ML in drug discovery. There is too much of hype. Of course, things are being made up and it's going on. A lot of pipelines and projects being tested, but not working everywhere. In some cases it works, in some cases it does not. So please be patient. It takes time, like Human Genome Project. So don't too much be overexcited. Someone is knowing AI. I don't know AI. So should I cop up with him? No, you'll be there slowly. So that is the first foremost thing I have to give. This is not a negativity of AI or ML. I'm telling you the fact, and you need not to worry too much. My slides will contain some of the pictures, videos, or contents taken from different articles and lectures or tutorials, and the copyright stays with them with the respective authors. This current slides is already available in my slide share. So all the slides, you can access it. I'll share the link in the chat after my presentation. And if you want to look at the hands-on videos, I already have a YouTube where recently started with during the lockdown, again, to teach young minds. So I have given a detailed tutorials on Autodoc, Modeler, PADL, QSAR, even molecular dynamic simulations too, including I have written a Python code for performing the complete molecular dynamics using GROMAX. And you can download that code and workflows from my GitHub also. Sorry, it is a self-advertising slide anyway, but I hope this will be helpful for many young minds. This is something which is very tricky and very alerting us. Of course, maybe not engineers sitting along for this meeting, but still it is alerting. It is saying that around 80% of the engineers are unemployable for the future jobs. That means AI or ML. So we have to start with this slide anyway. We need to know where we are. During this lockdown also, we have around 93,000 job opportunities for data scientists, still unfilled because they couldn't find the right candidate. So data science means it's not only for computer science people. Don't think like that. Data science can be for a mathematician. If you want to write an algorithm, you need to know a lot of linear algebra, matrices, probabilities, differentiation. I know you all will be feeling better. Why did I take then biology to avoid mathematics for some of the cases? Mostly they're on passion. So otherwise we have to have data. So data science is completely depending upon the data and how to interpret the data. And the data can be from anywhere: chemistry, drug, healthcare, it could be anything. So that's where we have to collaborate and find it out. So this is something alarming. So I hope you will take necessary steps if you want to go for interdisciplinary or multidisciplinary field. That's where we have to bridge the gap. You have to collaborate. It's not that I can do everything myself. Don't think like that. Never, ever possible. Even if you try to do that, it will take its own time. So why rather doing that? Initially collaborate, learn from them, then do it yourself. So you have to bridge the gap. Let it be anyone. When you're talking about a drug coming into the market, it did not come from a single expertise. A biologist cannot come with a drug. A chemist alone cannot come with a drug. Other pharmaceutics cannot come along with a drug. There are many components involved in it. So you have to collaborate and you have to bridge the gap. Whatever it is, hierarchy or anything it is, leave everything aside. It's research and focus to the result. We are in the current era of Industry 4.0 and we have to realize that. I'm not going to too much talking about it, but what is Industry 4.0 is Internet of Things, artificial intelligence, and big data. Now, as we are researchers, we have to generate data. One of the biggest drawback in drug discovery in AI, using AI or ML, is lack of relevant data. Data is there, but relevant data. Now we have to generate that data. When you see publications, there are a lot of uncertainties in experimental predictions too. There's no standard deviation reported. There is no mean reported because you would have carried out that experiment only once. Unless you iterate it for 3 times, how can we understand the error value there? I can understand it's because of the fund limitations or time limitation, but we cannot use that data because the experimental data itself is variable. We cannot have a confidence in believing that data. So we need to generate right data as an experimentalist for future usage in ML and AI. That's where the accuracy is going to increase. So we want to also look at distributed production, where we talk about blockchain and many other. Anyway, Industry 4.0, when I'm talking at the end of my presentation, I'll be talking about Bio and Pharma 4.0. So today's agenda, what I'm looking forward is, first, I'll talk about AI and machine learning. Don't worry, I'm not going to talk about any equations, something very basic to understand where you have to start. I'm not talking about any algorithms also, because the audience is mainly pharmacy and chemistry or bioinformatics and biology. And how machine learning is used in chemistry, what are the set of chemical data that is used in data science, and how drug discovery is utilizing these machine learning techniques to avoid failures and to avoid missed opportunities, something very, very important. And what to do next. Are we ready with AI or ML? Is it already there or not? So let's go through that, which is something feeling very bad for me to present, which is something reality for upcoming AI world era. So starting with AI and machine learning. let me pick up the word learning. So we have to learn, or the software has, or the model has to learn. Learning denotes changes in a system that enables a system to do the same task more efficiently the next time. So it's not simply learning, you have to learn, and when you're trying to apply it again, it should be more efficient. It's not that the same learning you have done, which was said by Herbert Simon. So this is exactly what is called machine learning. So it's a kind of black box, but you have to try to understand what are the different parts in that black box. So we have to understand and improve the efficiency of human learning. That's what we are looking at in machine learning. Improve the methods of teaching and tutoring people. Discover new things or structure that were previously being unknown to humans. That is something very important in machine learning. We realize and come to know many of the findings while training set of lot of data available. That is because of data mining and scientific discovery. And large AI complex systems cannot be completely derived by hand and require a dynamic updating to incorporate new information. This is where we have to understand the difference between information and knowledge. Information is like a tin of red paint, and knowledge, when that red paint is applied on a wall, you admire it, saying that, oh, very nice color, red paint, and it is being applied somewhere, useful. That's where you call it as knowledge. So whatever time you learn from books, whatever you learn from how to do tutorials, this is mostly information. Unless you apply it yourself on a problem, you cannot convert it as a knowledge, because from knowledge, you learn back something from your experiences. So learning new characteristics expands the domain and expertise and lessens the brittleness of the system. So that is called why we need to learn using these AI or ML. And reproduce an important aspect of intelligent behavior. That is, reproducibility and reliability is something very, very trivial for all machine learning models. Now, how can we specify this learning system? We will specify in three different parts. One is task T, performance P, and experience E. And we will specify requiring the following factors. Exactly what knowledge is to be learned, how this knowledge is to be represented, and how this knowledge is to be learned. So this is what three main aspects we need to know. So when you look at artificial intelligence, we are enabling machines to think like humans. But when you come to machine learning, it is training machines to get better at a task without explicit programming. Initial programming is required, but not later. And deep learning using multi-layered networks for machine learning, where we have a lot of hidden layers. We don't know what is happening, but at the end of the day, you get the outcome. Now, how AI is currently impacting our lives? Already it's there in smartphones. They know much better than what you're thinking. Even if you switch off your Wi-Fi, your Bluetooth, and whatever, NFC, whatever, still the Google can show you where all you went. So that is the technology, because you give S to everything without reading it.
Sir, you are not audible, sir.
Hello? Hello? Can you hear me?
Yes, sir, we can hear you. Please continue.
Oh, okay.
Yeah,
Someone
sir?
was saying, hello?
Vidya, madam, we are able to hear sir's talk.
Okay, okay.
Okay. Okay. No problem. So that's where we understand they are taking data with your knowledge because you gave a consent. So you need to, this is what the tricky part, risky part. You're not reading it, but they're taking your data. So you have to be very careful what you are addressing or agreeing to what. Social media platforms, e-commerce, navigation systems like Uber, Ola, Swiggy, Zomato, they all use AI ML to control and get up what is to be showed into your front page so that it is interesting to you. Banking and finance sector, automobile, autonomous vehicles like recently MG and even Hyundai, they all came with internet cars and even smart homes like Alexa and many more. This is all it's affecting or impacting our lives. It's a part of us now, Siri and all those things anyway, and Google Assistant. So we have to see how this is going to affect us in later stage. Now, machine learning is a branch of computer science which deals with system programming in order to automatically learn and improve with experience. For example, robots are being programmed so that they can perform the task based on data and they gather from sensors. And it's automatically learns program from that. So as you know, there are movies that also came in India, many about robots and others. Initially, it looks good. At the end of the movie, it's being parted up because of its risk and others. So the learning architecture also takes care of knowledge base. That's what we need, a lot of data for creating the knowledge base. And performer, the model to perform or produce a result. Critic, we need some feedback or to evaluate, and the learner. From that criticism, we have to learn. So this is also like a human learning process, right? So you have a knowledge base, you learn it, you try to perform it, and you get a criticism from person saying that this is not the right way, and you improve it. So this is exactly the same architecture for machine learning also. So the problem generator is to test the performance against it. So other paradigms are rote learning, interactive learning, induction, analogy, clustering, discovery, and genetic algorithms. I'm not going into each of this specific topic anyway. These are the different algorithms and based on data categorizations available like reinforcement, clustering, unsupervised, dimensionality reduction, classification, regression, supervised learning. Each one is around 3 hours of class actually. So don't get panic. We don't use all of them for drug discovery or chemistry related data. Mostly reinforcement is being used, regression is used, clustering is used. But there are many, many verticals available. So you need not to worry too much about that. Now, what are the types of techniques in machine learning? Supervised, unsupervised, semi-supervised and reinforcement learning, then transduction and learning to learn. And the 5 popular algorithms are decision trees, neural networks, probabilistic network, nearest neighbor, support vector machine. For example, in today's my presentation, I'll be talking about the application of probabilistic network, nearest neighbor, and decision trees mainly. The stages of ML is you have to build a model. How many of you know QSR? It's exactly the same, but the dimension is something large. You will have more data and more, modules for the model to improve by itself. So model building is one of the first stage. Then you have to do model testing and apply the model. So don't think that I should have a training set and test set, then the model is done. No, that is an old school QSR. We wanted to have training set, test set, as well as validation set, plus external validation. So you have to test your model being a third person. Until unless you do that, none of the QSR models will be accepted by ACS journals. ACS clearly mentioned, until these 4-step validation is being done, they don't accept, plus experimental validation. So that's where we have to look at. When you pick up around 1,000 papers of QSAR, less than 5% is reproducible. I didn't say, many of the reviews said that. So what is not machine learning? Artificial intelligence, rule-based, I'll be talking about rule-based inferences also. They are not machine learning anyway. So now let's see how machine learning is used in chemistry. As already being said, machine learning is overhyped or not. We have to see how far it is overhyped. To some extent, yes, and to some extent, no. So C and Ian, it's a journal from ACS again. So there they came up with a survey, around 151 people responded. 31.8% said that they agree that ML is overhyped, and 24.5% disagree to it. And many of them strongly disagree and others strongly agree. So it's like a kind of more of them strongly agrees or agree that machine learning is overhyped. And if you see the expertise, computational chemistry is only 7.3% from that responded. So we cannot simply generalize things, but still. And organic chemists are 23.3%. So when you come to a drug design or coming the drug into the market, the role of medicinal chemist is huge. Role of computational chemistry is very less. I'm not talking about this survey, I'm talking about generally. Once you design a compound in a computer, only a chemist can tell you whether this can be synthesized or not. Maybe software can tell you a probability of synthetic accessibility and feasibility. There might be hundreds of de novo methods. Until unless we can confirm this compound is synthesized or not, no point of any predictions. So that is a challenge that we are facing most of the time from computational predictions. So that's why when you submit to a journal with computational data, they will say speculated data. Don't feel bad about that word. They said it is a predicted data. We don't have any confirmatory data for take your paper for publications. So it is not what you did docking in 2005 and 2004. Now, if you're docking and just simply reporting the scores, they don't accept it because they don't want docking score. They want something beyond that, which we will discuss in a while. So what is a machine learning workflow in chemistry? We experimentalists try to start generating data, but ensure that you're generating the right and correct data. So that should be annotated data production. It could be from synthesis, simulation, screening, and sensors. And then we do a pre-processing, like cleaning, integration, transformation, because some data might be not correct. We have to remove them. All those things should be taken care based on statistical analysis. Then we apply machine learning models like SVM, deep learning, evolutionary algorithms, etc. And this training algorithmic learning, it is an iterative process. So don't think click a button, you get a model, that is a final model. No, you have to repeat it several times because the splitting of data set into training, test, and validation has different methods available. For example, clustering, Y randomization, and normal randomization and all. But it's not necessary that all three should work. Sometimes manual intervention is required. So that's why training algorithm learning is an iterative process. Don't think on a single time you're going to get the model. No, it's very difficult. So that's what we have to know, the expertise. That's what I'm a bit against this automated workflows of all these softwares, even though I'm coming from a software company. Because young minds should always understand the manual process of doing this each and every step so that you get to know what is happening in the background and you know what is doable and what is not doable. In the automated process, there are always errors. And they, on 30th October, I was giving a talk at BITS Pilani. So there was a speaker who was, his presentation was only to talk about errors in chemical data generated by softwares from databases and many. I was so surprised to see that even from ChemWell and PubChem also there are errors in the data. Even from experimental values, even from software generated data, there are many errors. With proof he has given it. So that is what we have to be very, very careful. Data reliability is something very important. So don't simply draw structures, put the values and start doing modeling. No, you have to ensure that is correct or not. And then you have to train the model. Now this is the place where there's a difference between machine learning and your normal QSA model. In a normal QSA model, you model it, you come up with an equation if it is an MLR, and then you finish it. But here in machine learning, this trained model, when the data comes, you have to add it up in order to increase its predictive capability as well as to increase the accuracy. So we have to iterate it with improving the accuracy. That is what it is, self-learning process is required. And don't think this can be done on a single click of a button. No, this is a several multiple process that to be taken care of. And finally, we can classify them, or based on regression, you can come up with a equation or model. Now, how different areas of chemistry data is used in machine learning. Let's look at the project-oriented data set. So you have a design of experiment, then you conduct your experiment, then you have a lot of data generated, and then you also try to model it based on predicted and measured data, and then that model is being generated. Now, we have to go for further optimization on validation, and we come up with proposals. If this particular region is there, if a particular group is there, we can have better activity based on this model.
Yeah, artificial intelligence.
Okay, fine. So yes, so we have to validate it and then we come up with the proposals. So now here the difference is, as and when the experimental data is being generated, your model also gets into active learning to minimize the number of experiments and maximize the properties. So the active learning is much more required as soon as when the data comes in. So that is where, when people are having a lot of experimental data available, you are the master, you are the king to do the machine learning because you have a lot of data with you. But unfortunately, we didn't have any practice in many places. We don't have any practice of storing experimental data other than what we publish. So that we should have been doing it, like in many research groups they do. Then framework for working with an in-house database. That's again, if a department is of a synthesis, like you do organic synthesis, they maintain an in-house database to understand full data of what they are like a mini sigma Aldrich or Molport. So you can understand what should be the new reactions or chemical hypothesis based on the historical reactions, failures, and success that you have faced from your in-house molecules. Now, moving forward, there are also data from simulations. For example, when you do ab initio calculations like DFT calculations for calculating energies or HOMO-LUMO, or even other calculations like MD simulations and others. So they have not been widely used because in very limited, it has its own restrictions to usage. So you can see here the values in electron volt predicted by machine learning is somewhat very close to DFT, but don't think that it is applicable for every, it has its own limitations. Then other is that when you work on public databases, you will understand some reactions, and using neural network, you can predict the reaction condition for this particular reaction, which could be the best catalyst, or which could be the best solvent or reagent, or at what temperature this particular reaction is very much scaled up, or better yield, whatever it is. So this can be also understood from neural network. So they are trying to learn from existing data to tell you or to predict and make humans to understand this is best for this, even though we already know from our readings. So that is the advantage of using computational tools, because we have a limited memory. Okay, 5 years back, if I read some paper, maybe 2 or 3 papers or points I still remember, maybe others not, but computers still remember. There are deep learning algorithms for solving different chemical challenges and the respective task also. For example, to design molecule, understand the molecule, especially properties, its orbitals and transition states, and synthesizing the molecule. How many steps taken care, like reaction prediction, retrosynthesis, reaction optimization. These all comes into deep learning algorithms. So we wanted to correlate the molecular property, the molecular composition with its related activity, with the training cycle, like predict, evaluate, optimize. So this is a cyclic process which is going on iteratively. And these are the schematic representation of the main components of atomistic machine learning. For example, it's not about predicting a model, it's about analysis and interpretation. So when there is a chart coming up, like snake plot is there, then there are clusters, the chemical spaces. What do you mean by that? By looking at that representation, what do you understand? So interpretation of data is also very important. I cannot simply say I'm a machine learning expert unless I can interpret the data which has come out of it. The data visualization is very, very important rather than the quantitative numerical data. So this is a recent publication from one of the pioneer in machine learning in cheminformatics, like Dr. Nathan. So he's very active in Twitter. You can see many of the time he posts about his models and others. So he was trying to do DNN and non-DNN models to see how far they are accurate and regression R-squares based on specific data. So he has taken data from Merck, from Kaggle challenge, activity data, TOX21 data, atom reactivity or atomic reactivity or toxicity data, even protein contact. And you can see different models are giving different accuracy and R-square values. I'm not going into it. I have given the reference at the bottom. You can read the paper much more. So we need to know which model or which machine learning algorithm to be used for specific set of data. Don't think that for me, neural network works, so it should work for everybody. No, nothing like that. It completely depends upon the data and what you're trying to achieve. Now we have to understand what the computers understand on a chemistry point of view. Machine learning methods needs a computer-friendly way to input the atomistic system. That's where when sophisticated software came, you just give the structures, but they do the back-end work to make the computers to understand what is this particular structure you have drawn. So for you, the structure will look like this. But for the computer, the structure will be converted to fingerprints or bits and binaries. And they look like this. So the issue with ML is arbitrary size and arbitrary order. And ideal general features are that it should be compact, unique, invariant, smooth, and fast. So this is what we are always looking at. And then we try to understand how far this data is reliable. That's what the problem comes here. When it is trying to write back, for example, machine learning algorithms are trying to predict and give us some of the structures. They have to convert from this particular bits and binaries back to the structure. That's where we are worrying about double bond. We worry about chiral centers and many more. So these are the challenges there available. But of course, they are updating the algorithms to overcome all these challenges in chemometrics and chemoinformatics. So as a global descriptor, this is for the structure, but for each different region, for example, this is a keto group or the CH group, these things are being defined in a different bits and binaries. So we have to be very unique on defining the descriptor for the machine learning algorithms to take care. Now we have algorithms and plugins and libraries to take care of this. So we need not to do it manually. It is automated. So now we understood all about what is this machine learning, where it is being applied, what kind of data is being used. Now let's go to the drug discovery part, especially understanding how to avoid the failures. The first thing being I'm talking to majorly a pharmaceutical technology department and related to an FDP for that. This is something very concerning for the common people. What I would really like is something to cure the high cost of medicine. What can you do for that? Can we, as a researcher, scientist, can we do something for this? Or do you blame government and research, regulatory affairs and demands and other interpolitics, whatever happening within the pharma industry? Whatever that is within our scope is that how to avoid the failures. Somewhere it has an impact to reduce the cost of the medicine. Few months back, I don't exactly remember, Sanofi got a rejection for their very promising drug by FDA. And they put a press release saying that around 1.15 billion US dollars gone. Of course, they're going to optimize it, come back, gone. How they're going to talk about it, how they're going to face it, actually. That's the story with many companies, not the same. It's they have to invest and they have to see how the success rate comes in. And then FDA approvals for NMEs and NCs per year also, you can see it's 20, 30, 11, 14. Not many compounds are getting, but the investment is huge. So this is what we have to really think of, how to reduce the cost of the medicine, how to give a social impact. Publications, patent is for filling our CV or our own, or sharing the research data to others. For me, publications are like sharing to other people what is positive and what I did is negative. So don't repeat that negative so that you can save time and money. When coming to patent, it's money. If you want to make royalties and commercialization, then only you patent. Otherwise, no use of patenting. I have a patent from US, which we were working on some types of insecticides, and it is already being commercialized. It is being taken up by a German company and they bought it. Of course, we are getting money. So this is what patent means. So we have to see the reproducibility, reliability, and how far it can scale up your research. That's where BIRAC, by Government of India, they give funding to scalable research. That means which can get into the market. It's not that always possible, but they look the vision, how far it is possible. When you're looking at drug discovery approaches, based on the availability of the data, you can decide which segment or which category of tools, approaches, or methods you have to apply. When you have an X-ray 3D structure, and when you have an X-ray 3D structures of ligands, that's where you have to do molecular docking. And please don't worry that many of them are reluctant to do molecular dynamics because it's too much difficult, lot of computing resources. But molecular dynamics is now mandatory. If you want to publish in a very good journal, I'm not talking about impact factors. I am against impact factors. Personally, I look to a paper, if it is good, how many citations you have. I'm not talking about self-citations. Being in a faculty-oriented program, I thought to tell about this to students, I might not say. So how far my papers got cited by others, that is the value for my research. Publishing in a high impact factor might be good. I don't say bad, but there are many other data issues also in high impact factor journal papers also. We have already seen that when we are doing modeling for QSAR. So what I want to tell here is molecular dynamics is something very much mandatory. You have to start learning it. Otherwise, you cannot publish in a very good journal, because molecular dynamics is the ultimate answer for whatever you have done in molecular docking, because it scales up in a solvated model with application of applying temperature, pressure, and then try to see whether the ligand is able to still stay within the pocket or it's moving out. Very difficult, takes more time, but yeah, research is always difficult. Now, why I mentioned the X-ray structures of ligands? Because your starting geometry confirmation is very important. I know most of us, including me, what we do is we draw 2D structures, convert it to 3D using softwares. Fine. But when you're converting to 3D, ensure that that is very close to crystal structure. For example, you have to do Hartree-Fock or high-level semi-empirical or DFT. You might say, sir, no time for that. Okay, then what to say? So you have to have a good starting structure. The reason I'm telling you, the confirmation of a compound in a stable form is different from the confirmation when it bounds to a biological complex, and both has to be stable. So we need to have a good starting structure. So that's where we call it a structure-based drug design. Software does whatever you give. They don't care what is a starting structure or not. Now they say software will take care of everything. Okay, as a scientist, I want to know what is taking care of it. I cannot simply believe that you are saying software takes care of it, right? We need to know each and every step. In other situation, you have a 3D structure of the protein, like X-ray crystal structure, and you don't have ligands, means you try docking everything with all database, nothing is happening. So then you go with de novo and scaffold doping. But the biggest challenge here is when you're doing this, please have a chemist sitting along with you. If unless he or she tells you whatever you design from fragment-based is synthesizable or not, no point of publishing any 100 papers or even coming up with high-level energy values or scores, because synthetic accessibility, feasibility should be checked. If you are a chemist, organic chemist, well, go ahead, no problem. But otherwise, please take help from your chemistry friend, so that, especially synthetic, so that before publishing, you ensure and you confirm that this is possible. Recently, I published a paper because now my group is completely focusing on longevity and anti-aging. So one paper we published and we couldn't find funds for further extension. So we discussed with organic chemist and they gave us a protocol, like the steps involved, and we put that in the supporting information. We didn't put it in the main paper. We said that we already discussed, this is a proposed protocol, and we published it. So that's what we have to come up. We are helping the next people to carry forward because we don't have money to do that. We are giving the proposed protocol step also. You can do it. So that is what we have to look at. So collaborate, talk to people, and put all possible data within it. Nobody is going to say no to that in supporting information. Now, the other step is ligand-based drug design. Very important at every stage. If you think molecular docking itself, I can find a drug. No, you cannot do. You will never find. You need also ligand-based drug designing approaches like Pharmaco4, similarity studies, QSAR, and many more. So in this case, you don't have a 3D structure of the protein, but you might have biological activity or the stable biologically active conformation of a reference ligand to screen your compounds. Let's say nothing is there. All these data is not there. Then you have to go for a combinatorial, like high throughput virtual screen. As Professor Mahan already mentioned, earlier days, it's just a key lock hypothesis. What I'm talking is in 1894, why Emil Fischer, to exercise a chemical action, a ligand interacting with a protein must fit into the binding cavity like a key into a keyhole. But currently, in 2020, this is only half of the truth, because multiple keys can now bind to the lock. And that is the situation. So you have to be very, very careful in understanding selectivity, specificity, and sensitivity of your ligand binding to the protein. Now, this single slide is something very favorite for me to explain what is and why is molecular docking. You have to, what is molecular docking? You're determining the optimal binding structure of a ligand. It could be a drug candidate, a small molecule, to a receptor like a drug target, a protein, and a DNA, and quantify the strength of the ligand receptor interaction. But when you're quantifying, it's not only with numbers, many more other factors. I'll address it in a moment. Now, if I want to explain molecular docking to my grandmother, that's what my professor used to tell. If you can explain your thesis to your grandmother, you're good with your presentation. That's it. So it's a layman way of explanation that we have to how translate science into a normal person. So we want to understand docking means where the ligand will bind. How will it bind? How strong it can bind? And what is the role of water or solvation and desolvation, water bound and water non-bound condition? What makes a ligand bind to the receptor better than the others? Translation and rotation of the ligands when it tries to form conformations. What is the role of torsions in docking? Did I mention anywhere about docking score? No. This is a picture that I have taken from enzymology book, actually. It's a really informative picture. These are the main factors that I would tell when you're writing a story about the molecular docking discussion part in your paper. You have to talk about the catalytic site or the catalytic function. You have to know the key amino acids, whether it is a catalytic triad or a motif region or an active site. Softwares might not help you. Database screening or database filtering might help you, and literatures might help you to understand this. Regulatory mechanism, whether your target protein is, whether your targeting is on the active site or on the allosteric site. The cleft detection. See, many softwares, most of them, 99% of them, they try to predict the binding pocket based upon the volume and geometry, where there is a cleave or cleft available. Is that enough? Because when you look at pharmacological parameters as well as biochemical parameters, we are looking at an upregulation or downregulation of a mechanism. So there we wanted to understand which is the key amino acids responsible for that. Of course, software will not be able to help that. Some softwares like ProSite database searches, ProSite will be helping you to understand the functional site, but still you have to read and understand what's the role of those amino acids. If those key amino acids are there in your pocket, that is your pocket. It's not that simply based on the largest volume, this is the pocket. So I cannot generalize it now. It's based upon your objective, why you're doing docking. It's a blind docking, go ahead with the pocket based. If it is very specific and you're looking to downregulate a specific mechanism, you have to be very specific on the amino acids too. Geometrical analysis, what kind of conformational changes it's going to happen when a ligand is going to bind in that particular pocket. Flexibility, whether it is an apo or a substrate bound region. Conservation, selectivity, specificity, and sensitivity. Like when you're working on multi-target drugs or on anti-cancer drugs or something related to that. Surface characteristics like solvent accessible area, polar surface area, what is a hydrophobic region. All these are very impact on the docking studies. And finally, interaction energy. And did you see anywhere docking score? No. Docking score is only used when you're doing virtual screening. If you have millions or thousands or hundreds of compounds and you want to select the best 100 or best 50 molecules, that's where you use scoring. Scoring is just to analyze the fitting comfortably of the molecule within the pocket, nothing else. Within that set of molecules that you have selected on scoring, then you have to do the energy calculation. Binding energy and binding affinity is what is a take-home message from your main docking program. So docking is several steps. It's not a single step and you can say, I have done docking. No. So you have to talk in terms of energy. Now, what is this energy and why it is used for? Because when you have the protein downloaded and loaded into any software, let it be any X or Y software, we have to do addition of hydrogens. When you add hydrogens, you have to think of direction of the hydrogen. You have to think of orientation, the tautomerization. So when you do minimization of your protein, nowhere it is said that you have to minimize your extra crystal structure before docking. It is a software's requirement. So in order to, why you are minimizing? Only to optimize the direction and addition of your hydrogen atoms to your protein, keeping all the other heavy atoms constrained or frozen. It's very much mandatory. I don't know how many of you have read about this. because we almost read only the tutorials and work on it. Recently, I have did a session on YouTube also saying about this. So we do minimization of X-ray crystal structure protein before docking only to optimize the addition of hydrogens. Don't optimize. You can optimize and also minimize if you are adding missing residues. For example, you did a further modeling. Of course, you have to minimize it. Then you have to optimize it, score it, visualize it, and edit. So docking is an iterative process if you want to design a new molecule, right? I'm not talking about virtual screen. I'm talking post virtual screen. You selected specific chemotypes and you have to now come up with a new molecule. Now, these are the desolvation terms we are looking at in energy terms. So energy terms is something very, very important when you're talking about docking results. Energy terms and affinity. Now, affinity, let me be very open with you. For example, when you're reporting autodock results, there is something called as inhibition constant. When I review many papers, I can see that they mention this value. I used to write to them back, please remove that number. Please put the range. A software cannot predict very accurately the affinity values. Energy terms are okay, still okay. So what you can do is put a range, whether it is between nanomolar and millimolar, still acceptable. Because software, if it is predicting 3.2 nanomolar, and when you do experimental, it might be 3.4 millimolar or micromolar. So we want to do something relevant, right? So try to put the range that is more reliable, and medicinal chemists accept it because they want to know range. If a compound is showing millimolar and another one is showing nanomolar, they prefer the nanomolar one. They don't know the value. They don't want to know the value, because that's where the difference between experimentalist and computer, because they always fight each other because they don't agree with many of the terms each other. So in order to go with agreement, we have to do this. So we look at hydrogen bonds, desolvation terms, clashes, and torsions. Torsions have a very big impact on docking. This slide explains about it. What we are, I hope you remember, I already told you, the conformation of the complex, binded complex, biological complex conformation is very important. Why I said is, when the compound is as it is, it's being stable, you are able to crystallize it. When it is bound to the protein, you should be able to crystallize it, right? That is where you say stable. So the crystal pack should be proper when it binds to it. So that can be statistically analyzed, understanding the torsions. So we are trying to compare with the Cambridge structure database to understand whether the torsions like this particular bond, for example, the green colored bond, is already available in CST. So they are frequently observed. So that part is actually crystallizable, or it is more stable. When it is showing yellow, they're very unusual, several times observed in CST. Red means they're very rare. That particular bond is very rare. So what it is trying to say is this conformation might not be the right one. You have to further optimize and do. I didn't say to change the elements or anything, might be possible. Otherwise, you have to do a very extensive conformation analysis to come up with a different pose. Another analysis is to understand the atomic contribution to your binding docking. So most of the time we talk about score and energy, but for a chemist or a medicinal chemist, score and energy is not going to help him, because for him, he wants to know which region of the molecule is so negatively contributing and positively contributing. So if there is a negatively contributing region, a chemist can always optimize it and improve it. That is what these kinds of packages is going to help you with that. So if these are called the coronas, the balls surrounding the atoms. So if you have a red corona, it's saying that that atom is negatively impacting the energy or the binding affinity of this whole molecule. So you can think of optimizing it. It is just giving a hint. It's up to the chemist to whether to make changes or not. So this is where we call it as 3D lead optimization. Larger the size, stronger the contribution. So that means what it's trying to say is, when the larger the red color corona, it is a positive energy. So we don't consider positive to be highly contributing. And the larger green color will be higher negative values. So they are more contributing to the binding affinity of this particular molecule. So the desirable properties, what we are looking at: biological activity, the related analogs, bioavailability, absence of toxic groups, irreversible interactions of biological target. And this is something that is commonly seen in autodock, right? I'm not against autodock. I have published many papers using autodock, even in very good journals with impact of around 14 also. So there is no against, but I want to tell you this because we usually see this. To overcome that issue, you have to increase the number of iterations and run. So we could always see inter and intra-ligand clashes when you try to do Autodoc. To avoid that, what you have to do is you have to extend the number of runs. So mostly you will do 10 or 100. So if any reviews come to me also, I used to tell them put it 10,000. Without 10,000, I'm not going to accept your paper, because then only all these clashes, everything will go off, because they do cluster analysis and then finally come up with the final. So they need more data. Just giving 10 or 100, they don't have data to cluster it, right? So this is what we have to understand. So and then racemic mixture. So in experimental, a compound with R form or S form will have two different biological activity. But how do you replicate in the docking studies? That also to be done. This all you have to visualize and understand. Don't think that a software is going to give you a number, R is better, S is better. Maybe there for automation, but your expertise is which is going to help you a lot. Now, this is something called chemical spaces. For example, you can, these are building blocks. One of the largest building block database is called NME. So what they do is they have a lot of building blocks using algorithms, using Pharmaco4 constraints. You can merge them and come up with novel molecules. But here, the additional advantage here is that it checks the synthetic feasibility. So when Merck tried it, Merck is a pharma company, Merck tried this, they found 85% of success rate. So they sent around 100 molecules, which they designed virtually using these building blocks, and they were able to synthesize 85 of them. 15, of course, not, because it's prediction, right? So this is how they do it. So you have what is the chemistry that to be taken, and you define the reaction that is the building blocks. There are millions of fragment space, and from there you design the model. Now, how this mapping is being done? We are not doing a structure similarity, because structure similarity, it's already very well proven that similar structures do not give similar biological activity, but it could give similar physiochemical property, but even different pharmacokinetic properties. So what we try to do here is we tried coming up with Pharmaco4 similarity. So what we did is we picked up a region and then did a Pharmaco4 similarity. And for example, for this region, we have around 0.61 or 61% of similarity. And for this one, around 75% similarity. This one, 91, this one, 99, this one, 89%. So local Pharmaco4 similarity is being checked, and we were able to, from a query molecule, we were able to come up with a very virtual hit from the fragment libraries. I mean, not fragment libraries, they are called as building blocks, with a global similarity of 80% based on Pharmaco4, not structural alone. So the success rate is here more because of the synthetic feasibility. And in order to check the synthetic feasibility, as I already told you, I'll be talking about applications of machine learning and AI. So this is another application where a retrosynthesis pathway predictor. I don't know how many of you are aware of SciFinder or Reaxis. This is something similar to that. This is coming along with StarDrop for academics. This is coming along. So when you give a structure, it tells you how far this is similar to the existing molecules being reported in a literature. So on your right-hand side, you can see the citation and the literature references where you can pick up the molecules. These are the applications of machine learning approaches. Now, this is one of another very important slide, which is the objectives of drug discovery. In this stage, I want to mention three different terminologies. One is hit, another is lead, and another is drug. Even in my master's, when I was doing a project on malaria, I put my title as drug discovery or drug design. But after getting to know much more about deeper, I thought that I made a mistake putting that title, because I cannot design a drug. Of course, I can do hit identification, heat to lead strategy, or drug-like design of one. That they are all appropriate words. So now you understand. I call, or usually we call a chemical compound to be a drug only when it finishes the clinical trial or it reaches the clinical trial. Unless, yeah, it is not. So we call it as drug-like or lead. When it comes to hit molecule, we are just identifying how they are tightly binding with the target and what is the potency. Those are the hit molecules. So when you did a virtual screening of docking and then you found some of the molecules, they are your hit molecules, right? Or lead-like molecules. And then you do pharmacokinetic properties like ADME prediction and toxicity. So ADME prediction, that's where I'm going through a few of the slides now. ADME prediction I have seen in many papers, they simply predict the number value, write something saying that this is a good solubility. That is not a ADME analysis. ADME analysis, we wanted to understand which compound we have to take from this, because sometimes solubility will be more, absorption rate will be less, and it might be toxic. Then how can we say that compound is more potent? Of course, it is potent, but not druggable. So we wanted to identify this chemistry with optimal balance of property. It's very important. You might think that I'm a biologist, why should I think about it? Unless you do this, you cannot get a drug in your hand anyway. So when you wanted to call a drug, you need to have optimal balance of potency, safety, that is toxicity, absorption rate, that is human intestinal absorption, metabolic stability. Your drug should be stable, right? When you're doing docking, you're already assuming that your drug has reached your target. But on a normal drug delivery mechanism, that administration mechanism, it has passed through many pathways, right? First, it will go through cytochrome P450. So you want to see whether the liver metabolism, whether your compound is very stable. If it is not stable, you have to see prodrug strategy. What is the solubility, permeability, stability of your molecule in the biological system? All those things you have to analyze. Unless then it will never reach your target as desired. So you have to quickly identify the situations when such balance is not possible. That's where we come to 3 conclusions: that fail fast, fail cheap. If it is not working, leave that molecule, start with the next one or optimize it. Only when confident you move forward with the next steps, because you have to invest a lot of time on and money on experimental analysis. Avoid missed opportunities, which I'm going to talk about in a while. Data overload is also a big challenge because we create a lot of data. Let's imagine that you have, forget about what is written on the screen. Let's say we have 10 different molecules and we carried out 10 different experimental assays. Each of these experimental assays are very, very important to make a decision whether this is going to be a drug candidate or not. So in order to find the best one molecule from a single experiment, it's very easy. You can say which one is top ranked and you can pick it up. Now you have to consider all the 10 molecules, all the 10 experiments individually or independently. And now you have to rank and choose the best 2 molecules. How to do it? Usually I get an answer, being this is not physical, I cannot ask you and get an answer. Otherwise it will take more time. So usually I get an answer saying that take an average or pick one property, leave the others. So we cannot do that because, for example, Lipinski rule of 5. Here, there it says molecular weight should be less than 500. And then we put a word saying that one violation is accepted. Let's say one property is already violated, hydrogen bond donor. And then your molecular weight is for one compound is 502. 500 is our limit, 502. Automatically it will say it is not satisfying the Lipinski rule of 5. Do you think that that molecular weight 502 should be neglected? Because it might be possible it could work. We don't know. We have to try. But initial filtering itself you thrown out or we thrown out. So those compounds, those are called missed opportunities. We shouldn't leave them. So we need to have an optimal parameterization to consider them also, to give them also some importance, so that they also come into a pool for research. If it comes in, it's good, right? So the data overload is also a big challenge. So where we get data from in silico, in vitro, in vivo. And now that's where we have to prioritize it. We have to give importance. We have to understand which more important. And we have to also consider uncertainty, because all data has uncertainty. Uncertainty in the sense, docking program, does it give you a standard error value or a deviation? It said it is prediction, right? Why there is no standard error value? When you come up with experimental value, IC50, did they repeat the experiment to give you the mean value or error standard deviation? No, right? We believe that they are the right or accurate. So uncertainty also is going to reflect on your predictions, and models also, machine learning models, very, very trivial, because experimental errors are going to increase or decrease the accuracy of models also. And then finally coming to selection. In most cases, we do select the molecules based on score or energy or some other value. But why we don't consider diversity? So it is already being said that when you're doing initial filtering of molecules, you have to give more importance to diversity than score or quality. Because we need more diverse chemotypes so that we don't miss any compounds while we go to the last filtering criteria. I'll explain through this slide. How you want to prioritize it. Let's say we have some amount of molecules, imagining the diamond is our best drug candidate. First, we did a potency test and some of the compounds being removed because they are not active. And then we followed up some selected molecule. We followed up with absorption rate studies and then metabolic stability studies. In each step, we are selecting and the number of compounds are getting narrowed down, and we expect that we are finally finding the best molecule. Unfortunately, based on the studies and reviews, we could understand that more than 90% this is not happening. Your good molecule might end up here, and finally you come up with a non-promising drug or molecule. The reason is, even though your compound is highly potent, it might not be having a good absorption rate, or maybe toxic, or we don't know about the metabolic stability case. So when you do a filtering one by one property, you might miss opportunities or miss compounds, which is really promising. If you're a chemist, of course, you can optimize and make modifications, and then, so that's where I always ask this question to chemists: how do you decide this is a derivative that you have to synthesize? Or how do you decide this is an analog you have to decide? Basically, for me, a medicinal chemist should decide on which interaction and on by seeing the 3D docking result, one. Second, understanding which region of the molecule is problematic. So you need to have softwares to help you for that. So here, this is where the rule is being applied. I was referring to rule inferences earlier. So here, what we are doing is, this is something similar to Lipinski rule of 5. So we can bring in multiple properties, give some threshold values, and rank the molecules. But additionally, what we have done is we have added importance level also. So here I'm saying the partition log P is much more important than ligand efficiency, molecular weight, and plasma protein binding category. So based on the importance level, this will be given as a probabilistic score. The good part here is, even though my plasma binding protein is low, even for the molecule, I mean high for the molecule, still that will be considered with the lowest score. They are not neglected or taken away from my data set. So this is a published example from CNS drug. So they picked up Pfizer candidates and they found only 60% of the molecule on an initial filtering mechanism or approach. But when they applied multi-parameter optimization, they found another 14%. So in this normal method, they missed 14%, but when they applied multi-parameter optimization, they found extra 14%. So multi-parameter optimization is a terminology I have seen in one of the syllabi of M-Pharm pharmacology in computer-aided drug discovery. So I don't know for others, do they have to learn or not. So they consider all these properties together to select the candidates. Now coming to ADME. What is ADME? I'm not going too deep, but just on a very explanation to a normal person, like where will it go? Distribution means where your drug will go, where distributed among your biological system. Metabolism, whether it will remain intact until it reaches the target. Absorption, will it get inside through, absorbed through your human intestine? And excretion, will it stay in and accumulate, or will it excrete at a proper time? So most of the time we can calculate volume of distribution, clearance, and absorption, including half-life and oral bioavailability. But we cannot predict the dosage amount and dosage frequency just with the structure. We need many more animal data in order to predict the dosage amount and dosage frequency. Now, how these models are all being transformed from a structure to these properties. We need to have a structure, then its corresponding properties called as descriptors, and then mandatorily we need to have some of the biological activity or some property from these corresponding structures, and then that will be your response variable called as Y, and then we use statistical or machine learning models to come up with the models. Now, most of the time we expect that I wanted to have QSAR with an equation. If you want an equation, you have to use MLR, that is called multilinear regression methods. But the biggest drawback of multilinear regression method is overfitting is an issue. Too much of false positives. And if we're using too many descriptors, and intercorrelated descriptors will be duplicates will be there. So it's a mess there. Statistically, your R square looks very good, but reliability, reproducibility, physical meaning, 0. So when you do QSAR and selecting the descriptors, please select only relevant descriptor. Simply calculating 3,000, 6,000 descriptor doesn't make any sense. Try to select the relevant descriptor for your kind of activity that you're picking up. When I was working in a US Department of Agriculture for mosquito repellency, so we have to do QSAR using their library databases. So what we did is we modeled QSAR, we got somewhere around 89 R square. But then when we try to correlate the descriptor with the repellency factors, they were not correlating with the physical meaning. I'm telling about the meaning or the significance. Then we checked relevant descriptors and we calculated some quantum chemical descriptors like vibrational frequencies and HOMO-LUMO and many others. Then we tried to correlate and we got a very good model with lesser error and lesser error in the sense better predictive capacity. Q square and R squares, RMSE were better. But the R square was only 82. But still we were happy and it was acceptable because not only statistics, both. So both has to be balanced. It's not only statistical parameters. And finally, you have to validate with an external set, not with your, so basically we say training set and test set, right? I am talking about an external set to validate it. And finally, you can predict for your activity and property. So there are several steps involved: molecular structure, descriptor calculation, classification, feature selection, model. So if you want to get more into the QSR, if you go to my YouTube, I have around 2.5 hours of discussion on QSR, much detailed explained. Now, the data set I told you, we have to split into 3, the training, validate, and test set. So we have to build models using machine learning models like GPR, Gaussian regression method. So GPRs are there, then random forest methods are there. TLS is something good where we don't have any overfitting issues, partially squares. Otherwise, random forest methods are also there. And finally, test with an independent set. Very, very important. Please try to make it as a practice when you publish any papers. Now, we have to also understand the domain of applicability. Madam, I have very few more slides, maybe 5 to 6. I think I can take a few more minutes. It's already 1:15.
Okay, sir.
Okay, thank you. Sorry if I'm making you feel hungry more. So domain of applicability is also to be checked because when you say this software predicts most accurate result, sorry, I don't believe it. The reason is, for me, for any QSAR or for any ADME predictions, I wanted to know what is the applicability domain, what is the standard deviation, what is the chemotypes in my training set. If they say that is proprietary, we cannot share, then I don't want to use that model. Because as a user, I wanted to validate my predictions myself. So the diversity of the training set defines a domain of applicability. If my training set contains all different chemotypes or chemical class of compounds, then the diversity is well defined. Now, if I say software A is predicting ADME, software B, C, D, E, and many online platforms are predicting. How can I, and for the same structure, I'm getting different values. Why is it so? And which one will I believe? I don't know. Because I will believe maybe someone said this is best, I'll go for that. No, you have to understand what is the standard deviation. So that's what I'm trying to talk about here. So imagine this is your training set. This within this boundary is your training set. That means a chemical class of compounds in your training set. I have given a compound for prediction. After doing the prediction, when I did a similarity analysis both on property as well as on structure, I found that my structure is fitting within the training set. Here, the standard deviation will be less. So I have more confidence in relying or justifying that this prediction is almost good because it's within the applicability of domain. If it's slightly outside of my domain, it's okay, because standard deviation will be there. Still, you have to ensure that the prediction is not that great. If it is too much far away from your model, then you shouldn't ever consider this prediction because it is not at all fitting with the applicability domain. So this prediction is not reliable. That does not mean that the model is bad. That meaning is your class of compound or your type of chemical class of structure is not present in the training set. That's why the deviation is small. So how far diverse we can have? There is a limitation in models, right? So we have to use multiple tools to understand which one is giving the least standard deviation from the training set, and that you have to pick up. It's not that always one single product or tool can give you one-stop solution for everything. Never, ever. The challenge of applying deep learning also. So recently, as I already told you, we have more challenges coming up in QSA because of unavailability of data and also more errors in experimental data. So the compound bioactivity or property is very sparse. Big data in pharma is not very big at all, which I told you from the beginning. Biological data is very much noisy because experimental variability is already there, 0.3 to 0.5 log units. When you're trying to publish in JMedChem or a little bit more expected journals, they look at the experimental variable. They ask you how many times you have repeated this experiment. So that is the expectation they're looking at. And that's exactly the expectation of a pharma company also. When you're patenting a molecule, why, if your compound wants to get commercialized, they will see all these things. How much times you have repeated? What is the confidence score? Everything they wanted to know. Otherwise, they cannot take it up. And how can we learn from this experimental data to make better predictions for compound bioactivity? So we applied some machine learning approaches and deep learning approaches to overcome this challenge. So for that, what we have done is, wherever you see light colored squares, they are the missing data for biological activity. But still we get the prediction, right, or overall imputation. So prediction input features to predict one or more property values for a compound. And imputation is the process of filling those gaps in sparse experimental data using the limited reserve that is already available. So what we're trying to do is, when you have a limited experimental data available for similar class of compounds or very close related chemotypes, we are using imputed assays. We are able to generate these data automatically using machine learning models. And so that we are able to come up with a package accordingly to available. So which several being published and already talked about, the proof of concept on benchmarking dataset, which is being checked on 13,000 compounds with 159 experimental assays, and application of heterogeneous data with Constellation Pharmaceuticals, where 2,453 compounds were tested with 18 endpoints. So here imputation means we are adding the missing data for the completion of better predictivity. So it is working the other way around also. So if you have sparse data in experimental, there are machine learning models learning from the existing experimental data and predicting the values for the other assays. So this is also possible. Now, this is where we have to completely understand a drug discovery means is a multi-interdisciplinary, whatever you call it, because we have to closely work with an informatician, closely work with a chemist. They have to talk to each other with the data. And visualization is very, very important. That's where it gives you more knowledge and information. And the chemist and the computational modeler also has to talk to each other in the same terms, because chemist has to see the docking result in the 3D form to understand where are the explorable regions in the compound where I can still make derivatives, so that there is some space left in the pocket. So simply not making derivatives, but making derivatives which could fit into the pocket. And also modeler is helping to understand which region of the molecule is problematic for ADME, so that the chemist also understands this is the region that I have to optimize, not all the region. Now, if you ask me what are the success stories of CAD, there are many success stories, more than 112 molecules which has gone to phase 1, phase 2, phase 3 level, even to clinical trials level, I mean the final stage also. So if you want to more read about it, these are the two main reviews. I have picked up these molecules, and also a very nice blog, practicalfragments.blogspot. Every fortnight they post based on fragment-based drug designing, what are the success stories in drug pharma industry. So this is really interesting where you can learn more about what they have taken the approach. Don't think that just with docking they have done many more analysis what to be taken care. So docking is the first good start point to find out the hit. And after that, to define it as a lead molecule, you have to do PKPD studies, that is pharmacokinetic and pharmacodynamic studies. And then finally toxicity, and then you move forward. There are open source tools like 9 Notes. I have in my GitHub also, you have these workflows available. Many things you can do, Pharmaco4 screening, then docking, RD kit, descriptor calculation, QSAR, Lipinski rule of 5, many more, even modeling, fragment, like the Pharmaco4 modeling, all those things can be used in 9 mode. So we have to explore what is available. Now, when coming to open source, I have to tell you, what is the main difference between a paid or a commercial software and open source? For me, main 2 reasons. One, on open source, you have to post it in a forum. Nobody is going to sit there and write an email back to you saying that this is how to be done. Nobody is going to sit and help you. It's all you have to take care. Second, regular updates and being tested and tutorials, all documentations being pakka and everything will be there. But did I mention anything about accuracy or validation? No. Both are equally accurate. Only thing you need to know what to apply, what not to apply. When you say about commercial tool, there are tools where you cannot put metal ions into it. Even in Autodoc also you cannot. In commercial tools, there are tools where you cannot do electrostatic field calculations. Open tools, yes, there are still they can do. So A, B, X, Y, Z, whatever softwares you use, please try to use, as Mohan sir rightly said, you have to use multiple tools and come to a conclusion on one. Don't think that I told you A is good, you have to use A. Because for me, A is good for my biological system. But your biological system might be having different composition of packing of crystals. So it might be different. So we cannot fit everything into a single one. So you have to evaluate it and make a decision. Here is another workflow available for environment toxicity prediction, for carcinogenicity, mutagenicity. This is also available in my GitHub where you can use it. So just give the SDF file, it will predict all these environmental toxicity prediction, give you as a table. And site of metabolism, like isoforms of CYP P450, like 3A4, 2D6, and 2C9. And when you're using machine learning, especially you need to know Python. So for Python, the easiest package that you can take is Anaconda, where you have many of the libraries comes along with it, Numba, Pandas, and many more. I think Dr. Malliga will be talking about much more tomorrow, as she's also having a hands-on with, I don't know, tomorrow, day after. So I'm coming to the conclusion of our presentation. What to do next? So we are in the era of pharma or bio 4.0, where we need to understand the importance of drug discovery and research scaling up, not publication. I'm talking about scaling up. Many of us are now talking about entrepreneurship. Many of us are talking about startups. So I do have 2 companies, which one is already 5 years old. Another one is around 8 months old. So this is also like a spin-off from our research. So we have to understand model outcome testing, collaboration with stakeholders, mobility-driven real-time research data exchange is very important. So also please keep ethics very important. When you collaborate, you have to acknowledge and give the research ethics. This is something very important. That gives you more value for more collaboration and future growth in your research area. Now, these are the digital transformation that is being expecting. And this is a picture that I have taken from, I hope you all know Foldit. So there is a game called Foldit, which is for protein structure solving mechanism. It's a very nice game. You will get ranks and scores and all. You could try it if you're playing many other games, because this is all to understand the van der Waals, electrostatics, and the stability of the protein 3D structure. So they are also applying virtual reality and augmented reality, getting into the protein and then trying to solve the structures and many more others, including image analysis and many others. Then we are also looking forward to 3D printed structures. I couldn't imagine, but yeah, we can think about 3D printed drugs too. Let's see, hope for the best. And even personalized medicine, which we already many are working on it. But this is something alarming slide for this whole presentation. AI looks everything good, fancy, everyone is admiring, everything is good. For today's condition, it just looks enhanced, okay? But 2040, when artificial general intelligence is coming, which is trying to expand more into humanity, our job is at risk because they learn themselves and then do it. Why do you require human expertise there? And when it very much super intelligence, when this AI is much more super, much more efficient, then humanity is at risk. So this is taken from an organization who works on AGI. From there, I have taken this picture. So what I'm trying to tell you from here is, leave something for us, whatever you're doing. Everything is good for the betterment of science and to avoid failures, mainly to avoid failures so that we can reduce the cost and many other factors. But we have to have a balance so that humanity won't be at risk. So to conclude, initially consider when you are trying to select molecules, give more priority to diversity and less priority to score and quality. And as the size of the lead molecule reduces, the funnel gets narrower, then you give more importance, 75% of importance to score and less importance to diversity. So this is how you have to consider where the enrichment factor is very important. I don't know how many of you are reporting enrichment factor in your virtual screening data in publications. Recently for drug discovery hackathon, we made it mandatory that they have to report the enrichment factor. So there is an equation that you can calculate the enrichment factor based on total number of compounds considered for virtual screening, about how much selected, how much is reported, all these things. So this is something which really helps a person evaluating to understand what is a diverse set of molecules or number of molecules you have considered for your research. It's not that simply 10 molecules being selected and doing docking, enrichment factors won't be great. So you have to start practicing enrichment factors. And also understand good synergetics between human expertise and computation tools are very important. Don't think that computational tools can do everything. That's where we always say GIGO, G-I-G-O, garbage in, garbage out. You need to know what is the structure you're giving to the computer software. Otherwise, they don't give you a right result. So if the protocol and the data is proper, you are more confident in relying on the result. Try to avoid missed opportunities. Don't use unnecessary filters or Lipinski rule of 5 unnecessarily in all your objectives. Lipinski rule of 5 is mainly used for oral bioavailability. Why should you use for something else? So don't use it everywhere. Maybe someone has already done and you are following the same thing, but check whether it is really required for your disease target or objective. Understand the significance of parameters or properties. If you're changing a specific parameter in an algorithm or a model, how it is going to impact. If the pharmacokinetic properties of a compound is increasing or decreasing, how it is going to impact. hERG liability, I don't know how many of you know that, it is the cardiotoxicity, hERG liability. So it's something very much mandatory, even though when you're reporting any compound, a probable drug candidate. Evaluate and decide a tool and or approach. Don't simply pick up a tool and then do your research. No, evaluate similarly. So as Professor Mohan already mentioned, pick up a paper, try to reproduce it. It is not necessary that you have to use the same tool because sometimes it might be paid, you don't have access. You might be using another tool. Of course, you cannot reproduce the same date, same number, not at all possible because it's a different algorithm. But you will still see the same trend, still see similar interactions. That is what we are looking at. So whenever you're using a tool or approach, even if you're using your molecule with a specific tool, try to do co-crystal template docking. You have to understand whether my software is able to reproduce the same interactions, what is already reported in the co-crystal or bound complexes. That's where the direction of hydrogen is very important. I don't know how many of you were aware of that. Depending upon the direction of the hydrogen, the RMSD value reduces. If it is different, it increases. Now you'll say the software will take care of it. Come on, we are talking about science, not about software. And also check the reliability of data used. And that is something very, very you have to check. So keeping all this conclusion very important, I would like to conclude my talk. And thank you so much, everyone. And sorry for taking another 15 minutes of your lunchtime. And I'm happy to take any quick question because I know already there is a shortage of time.
Thank you, sir, for your excellent presentation. There is one question in the chat box. Is there any way to calibrate the software to check its efficacy and precision?
To check the efficacy and its prediction, the calibration can be done only with experimental values. So if you have a set of experimental data with you, try to use a software, try to predict it, and then try to correlate and see. Now, let me be very open here. When you're trying to do docking, and when you try to do a bioassay like predicting IC50 or ED50 or whatever, And then don't try to correlate between IC50 and the dog score or binding energy. No, they are all both in different terms. So please do not correlate them. What you have to see is in the binding affinity prediction or the binding energy prediction, if the docking molecule is lesser, here also, better, here also do you see whether it's a scaling up in the activity. The trend you have to see. So calibration can be done with a comparison to experimental data.
Okay, sir. Participants, any other questions? You can please post in the chat box.
And I have posted the link where you can find this slide. The today's presentation slides.
Yes, sir. Sorry, I think there is one more question before I conclude.
Okay, there was a question on the research internship. So we do take 2 interns per year. There is a selection procedure, and you have to bring up your project, discussing with your faculty. If it is a project internship which is more than 5 months, it's completely free. We don't charge you, and you can use all our tools as well as open source. So when you come up for project, you get trained as well as you do project. So you are getting trained with theories and softwares and practicals, everything through your project. So what is the requirement of the internship? You need to have a lot of patience for reading. Read. Second, every week you have to present. Every week you have to present. So a lot of patience on preparation of presentations and reading. That is what the energy you require for research internships. And you can email me if you're really interested with your CV, 1-page research proposal, statement of purpose, and a recommendation email from your faculty.
Thank you, sir. That's a very useful information for our scholars.
Yeah.
So to conclude this session, sir, your session was excellent, which included the basic difference between artificial intelligence, machine learning, and deep learning. And the most important aspect was how to utilize machine learning in chemistry, because before this session, all of us were having lots of doubts, whether we are using machine learning, whether we are using it in our CAD, whether CAD is a part of it or not. There are lots of ambiguity, but your presentation made it very clear. The DL algorithms for solving different challenges, importance of data visualization, descriptors for chemistry, importance of starting structure, which you have explained in a very simple words. That was very, very interesting of your presentation. The concerns about molecular docking beyond binding energy, because many articles focus mostly on the binding energy. What else we have to focus beyond it? That was very excellent. Challenges of data overload, especially in the biological context, the data overload importance, you have explained it. Relationship between the descriptors and validation in the QSR program, and the most important one, way forward of using machine learning in the drug discovery process. It was excellent. Thank you very much on behalf of our department, Institute of Pharmaceutical Technology. I thank you very much for taking your time to be a resource person in this FDP. Thank you, sir.
Thank you so much, and best wishes to everyone.
Thank you, sir.
Thank you, sir. Thank you very much. We also expect same cooperation in future also, sir.
Absolutely, absolutely. Happy to do that.
Thank you. Thank you very much. We'll be in touch with you, sir.
Sure. Thank you. Always pleasure.
Thank you.
Madam, shall we announce afternoon session?
Afternoon session at 2:45. Yeah, you just announced that.
Participants join the afternoon session at 2:45. Thank you.